at the click of the button, but you just have to post a message to the chemist and it is being delivered at your doorstep also. But how it evolved. When we talk about maybe in the year in around 16th and 17th century, when people in Europe they started using opium, which is a milky sap from obtained from the poppy plant, and it was used to treat pain. And later it was realized that this mixture of opium was mixed with alcohol and few herbs, and this was used and this was named as laudanum. And this laudanum in Europe was used to cure everything, whether it is a simple cold or a diarrhea or even to fight against black plague. This was the only medication that was being used. Whereas in India, we know that we had a very rich system. And today also, even many, because there are so many researchers, scholars who are working in this domain where they are working on the Ayurvedic medicines, trying to extract and give a more scientific uh, justification that why we are using that particular medicine, because maybe that information is not it is there in our literature, but maybe because language becomes a barrier, and that is why we are relying on the research. And in that is why maybe the emphasis is nowadays laid on reading more, learning Sanskrit, so that we are able to read our literature and explore and extract the information that is embedded in it. And in fact, in the year 1804, a German scientist, Friedrich, he extracted a main active compound from opium, which was called as morphine. And this morphine, which is morphine, it is named because this is the Greek god of sleep. And this medicine was used to cure from pain. And nowadays, even till date, morphine is being used. And then it just started. After morphine came quinine, which a cure for malaria in the year 1820, followed by paracetamol in 1877, though the commercial use of paracetamol started only in the year 1952. And then came aspirin in 1819 from the bark of willow tree. I think maybe I would like to share one more thing with my students here, that if you look at it, the first medicine that was invented in Germany. And if you look at the literature of Germany, you will realize, and even today, if you look at the structure there or the institute, if you look at their universities, in Germany, more than 14 universities, even till date, they offer a course in Sanskrit, because they feel that Sanskrit is not a language of worship only. It has so much of science hidden in it. And that is why they are offering courses. And the first discovery, and all medicines generally, they were discovered from plants only. So maybe we say that it has been even often said that maybe Germany is carrying the legacy of Indian literature and culture also. And then we realized that in India, because this is how the world was progressing, but in India, the first synthetic medicine was developed in 1922 by Dr. Upendranath Brahmachari. And he did say the drug that he invented was urea stibamine. And this was used to cure black fever. And black fever, it is also, we call it as visceral, because and visceral leishmaniasis. And this was, and because of his invention, in fact, he was nominated for Nobel Prize in Medicine twice, but somehow he was not given. Had it been awarded to him, he would have been the first Indian scientist. And when we are here, maybe so many things are happening simultaneously. When we are celebrating Azadi Ka Amrit Mahotsav, celebrating 75 years of India's independence, we must acknowledge the contribution of our Indian scientists, our researchers who actually contributed. But maybe so many things were happening. So even if he did so much of work, we many people are not even aware of this. When I talk about my students, and as a teacher, we never lose any opportunity to speak with our students, and that is why I thought that I should share these things, these insights with them, so that they should understand that how things developed. And when we are here today, we are using advanced technologies and so many things and so many medicines, everything is available, but it is not the end. Whenever we say that research, it is always a beginning. You will find that so many new things are coming up day by day, and we have to continuously engage. And this is one of the prominent field where when we are working, it not only because we say that this is one of the noble cause towards society, because when we are developing a drug, we are not only maybe you are able to fetch fame, fun, money, wealth, all other things come side by side, but what you are actually doing is you are bringing in faith and relief amongst billions of people who are actually relying on this. So when we get the opportunity, so we thought that this is one of the platform through which we can connect and motivate our young minds and inspire them to work in this field. And I will request all my participants that this is certainly a golden opportunity for you all. So don't lose this. Grab this opportunity. Whatever comes into your mind, whatever query you have, you just post it in the chat box, and we will ensure that all your queries are answered well. And we want that India, because if you look at the Indian pharmaceutical companies, we know that for the production of generic medicines, India is among the top companies. Indian pharmaceutical companies are among the top. And in fact, in terms of the production of medicines in volume, we are ranked third in the in the world. So, but there is still a lot that needs to be done, and that can be done only when we have sufficient means to motivate our young researchers, our students. And with this thought only, this particular conference was conceptualized. And I can see that still the number, we started with 201, and right now the number is 212. And by the time we get the opportunity to hear from our eminent speakers, this number would increase many fold. So, and this, I take this opportunity also to congratulate the chemistry department for coming up with this thought of having this e-conference and using this time when the students, even if they are at home, using this time in a very constructive way, motivating and inspiring our students. So thank you so much for making me a part of this conference and giving me the opportunity to speak and share my work. So thank you so much. And on the behalf of Maitrey College, I once again extend a very warm welcome to Professor Prasad and Professor Saraswati. Thank you so much.
Thank you, ma'am, for your inspiring words. It is now my pleasure to introduce you to the chief guest, Professor K.N. Saraswati. She is the chairperson of our college governing body. We are delighted to have you with us today, ma'am. Ma'am is presently professor at the Department of Anthropology at University of Delhi. She did her PhD from Department of Anthropology in the year 2000. She did her postdoctoral fellowship from Molecular Genetics Laboratory of Albert Einstein College of Medicine, New York. Her areas of interest include molecular anthropology, genome diversity, and molecular medical genetics. Professor Saraswati is a member of several national and international professional societies and has published over 115 research papers in journals of national and international repute. She has authored 7 books and handled over 15 research projects funded by DST, DBT, UGC, ICSSR, and ICMR. Ma'am has guided over 20 PhD theses, 8 MPhil dissertations, and co-guided 7 MD dissertations. It is an honor to have you with us, ma'am. I request you to kindly address the gathering.
Thank you so much for this kind introduction. A very good morning to the esteemed guest, Professor Ashok K. Prasad, Dean Science Faculty, the principals of Maitrey College, Dr. Haritma Chopra, Dr. Lata, the convener of the present international seminar, the teachers of Maitrey College, the staff of Maitreyi College, and also all the students of Maitreyi College and the participants. It's a pleasure to be here in this platform, and I thank Dr. Harithma to remember me at this point and call me here to say few words. And most importantly, I would like to say that I have been associated with Maitreyi College maybe for the past 1 year or so as a member of their governing body. And I should tell you, I think this is a point where I should point out this, that being associated with Maitreyi College is a good experience, a lovely experience, because this is a college. I call it as a happening college, something or the other. I've been observing for the past 1 year, like in spite of this pandemic situation, they are doing something or the other. Every time some art competition, some seminar, some workshop, continuously some fair or the other, the college is engaged. And it is surprising that all the college teachers, they do cooperate so well with the principal and they work in tandem. It's really wonderful to be associated with such a nice college and people of Maitreyi College. And I really enjoyed my association with this college. Now coming to the today's international conference title, that is drug discovery and development. When we talk of drug discovery and development, of course, it is a very, very important topic as on today, especially after facing this corona, where we didn't have a drug to face this corona. So at this juncture, we really feel this is a very wide and a very good research topic. And having such seminars with such contemporary topics definitely will enlighten the young minds who are in graduate level and undergraduate level students. If they are exposed to such topics and such talks, definitely the research oriented minds, I think, will be developed. So it is very important to have such things at college level. Now, when we talk of drug discovery, Dr. Harithma elaborately talked about the importance of drugs and how the history goes on. And it was very interesting talk, of course. I'm not going to those aspects, but I would like to talk about something other than drug, which also have to be looked into while developing a drug and, while, discovering a drug. So one important thing is when we start off this drug discovery, it starts with a disease. You need to know what is the disease first, and then whether the disease is genetic or whether it is non-genetic or infectious or lifestyle. And after that, we have to understand the pathways, biochemical pathways involved in that disease, and identify the target molecules. Once you identify the target molecules, then you work on in vitro animal models, and then you come up with certain modifications in the drugs, and then you go for clinical trials and then get permissions from the government, and then it reaches the hospitals and clinicians, and finally it is prescribed to the patients. So this is a whole process which looks as if it is a simple linear procedure, and then it has got more of biological component. But however, we should realize that this is an area where the expertise of different domains have to be included. For example, clinicians are needed here. Biochemistry people are needed here, statisticians are needed here, public health personnel, and many more other fields are also needed in such research endeavors. Now coming to one of famous scientists, Archibald Garrod, maybe 150 years ago, what he said was, like one man's meal is the other man's poison. So you also can take it to drugs also. One man's drug may not be suiting the other man. So this is the vision a person had 150 years ago, and he was the Archibald Garrod. He's very famously known for Garrod inborn errors of metabolism. He was the first person to bring out the heritable nature of certain diseases. After that, only Mendel came up with the dominant inheritance, recessive inheritance, so on and so forth. But he was the person who talked about inborn errors of metabolism 150 years back, and that too among human population groups. And then this one man's meal is another man's poison. It is now coming to be true, because after Human Genome Project, people have realized that genes do not behave differently in different environments. So Francis Collins, NIH America, the chairperson once upon a time, he said, genes load the gun, whereas the environment pulls the trigger. So these are two important. When I was a student of MSc in anthropology department, when I was studying human genetics, my teachers taught me there are categorically 3 types of diseases. First of all, 2 types of diseases, communicable diseases and non-communicable diseases. And second is, within non-communicable diseases are those diseases which have a genetic background, and communicable diseases are infectious diseases. And then there are certain lifestyle diseases. So basically they could categorize things. But after Human Genome Project, there is nothing like such categorization has vanished now. There is no such categorization. Everything, every disease, every abnormality, any sickness has both environment and the genetic component. And even if it is an infection, corona infection also must be having some genetic predisposition. You must have observed it in single family. There may be 5 members, 2 are affected and other three are not. This could be because of the 2 being more predisposed to corona infection. So that means genetically some people are predisposed. Basically, I would like to tell you is that here now in these contemporary days, we cannot say this is a genetic disease, this is an infectious disease. The question is, how much is the contribution of genes and how much is the contribution of environment? In certain diseases, genes play a major role. In certain diseases, environment plays a major role. And then next, when I go beyond this, like when we talk of these drugs and discovery of drugs and all these things, genetic architecture and epigenetic architecture is widely ignored. So when I'm talking about the gene environment interactions, when during the drug discovery trials or research, this genetic and epigenetic component of human populations needs to be addressed, because genes are there and genes affect the gene expression. And epigenetic is a Lamarckism comeback. Everybody asking, if you are a science student, everybody must be knowing what is Lamarckism. Lamarckism is the inheritance of acquired characters, and it was absolutely refuted long back. But now it is coming back in the form of epigenetics, where something around DNA happens, which is heritable and also reversible. So this is something very, very interesting. So epigenetic and this epigenetic alterations do affect gene expression like the mutations do. So what I mean to say is both epigenetic and genetic architecture of populations or human beings has to be taken into account, and there must be more research, tailored drugs, individual specific drugs. So these are the new concepts after Human Genome Project we are getting. For this, I will give you a nice example which we have been studying throughout our BSc, MSc and PhD. Everywhere we do G6PD deficiency. This is an X-linked genetic disease. And when you say X-linked genetic disease, obviously it is expressed in males in hemizygous condition. So when we talk of G6PD deficiency, if the mutation is there in the X chromosome, that particular disease expresses. But it is surprising that this disease is not expressed as just like that. The males are as normal as everybody. They're born normal. They can stay normal. They can die normal. No problem at all. But that expression of the mutation is seen, observed only when there is a when there is some sort of what do you say, oxygen tension exposure. Like if they are going to high altitude, they will undergo hemolysis. If they use certain drugs like sulfur drugs, malaria drugs, they will undergo hemolysis. If they are exposed to certain genes, they are uh, having hemolysis. And if the hemolysis is not addressed immediately, it may even lead to death. That means here genetic mutation is there, but the genetic mutation is silent as long as it is not exposed to the required environment. So gene and environment interact to give a phenotype, a deathful phenotype, which is very, very dangerous. So this actually is happening to most of the genes in the body. Some are so fetal, they are not able to survive even to infancy level. And some are so mild, we are not able to catch them. So this type of issues are to be addressed. I don't know if they're already addressing all these things. So you need to take care of all this. Second important I also would like to say is environment. Now, what is environment? Environment, we think it is, like uh, air pollution is one of the components and environment. Now you talk nutrition is an environment. And apart from nutrition and air pollution and many other environmental things, we also need to consider about the culture of populations. Culture is something, that is passed on from one generation to other generation. So that cultural attributes which you are thinking, that a vegetarianism is a cultural attribute, though it affects your biology, but it's a cultural attribute. You are a vegetarian, then she's a non-vegetarian. Such cultural attributes do make some impact if they are exposed to, if you are out of your culture. So there is some sort of stress on you. You are, suppose I am from a very orthodox Brahmin family from South India, and if I am put into a Delhi and I am asked to eat with people with, I mean, it's not me only, anybody for that matter. If I am asked to eat with people with non-veg, vegetarian, it will be difficult for me to adjust in those situations. Even by force, I may have to do, but my cultural, brought up doesn't make me comfortable in new situations. These are the issues that also have to be taken care of. And so there should be, there is a lot of paucity of data, especially in Indian context. It is a developing country where, recently till now, we have always been thinking that rural populations and tribal populations, they do not have cardiovascular diseases. But now there is an upsurge of these diseases even in rural and tribal populations. Our studies recently in Bhil population also, we have found very high frequency of hypertension, though they are not obese, but there is hypertension. The route for hypertension is different for them. So if normal hypertensive drugs which are targeting the metabolic pathways are given to those people, it may not work. You need to look for different drugs which will treat hypertension in those populations where the route for hypertension is likely to be different. So for that reason, we need to first create a database where we screen the populations for various diseases and their risk factors, and also and document them so that they are available for public health interventions. This is very, very important. Even this is an important thing, that drug development and discovery and development people who are doing research on this, these things are also have to be kept in mind. And then there should be awareness programs. Prevention is always better than cure. Everybody knows, a famous saying, everybody knows from the beginning. So drug is something that you give only after the event happened, adverse event happens. Now you try to reduce the effect of the event. And sometimes you cannot complete diabetes, you cannot completely cure it, you can manage it. Hypertension, you cannot cure it, you can manage it. So these are the drugs which are coming into place only when the event has already happened. So there are ways which can be used to prevent the occurrence of yoga and everything. Of course, I don't want to go into all those things, but there are ways to prevent. The prevention is always better than cure. This doesn't undermine the importance of drug discovery. Of course, drugs should be there because there are certain life-saving drugs. You cannot go get away with them. In spite of very good lifestyle, very good genetic background, you are prone to certain diseases, and for that, drugs are the only option left with us. And now, I think especially in Indian population where diversity, as I already told you, there is a huge diversity. So it is not, I mean, anything that works in a caste population of North India may not work in the caste population of South India. And anything that works in caste population may not work in tribal populations. They have different gene pools. They are exposed to different environments. So these are important things, and these things are also to be taken seriously. And people, we need to deliberate upon all these things. Maybe Dr. Haritma, if you agree, we can have another international seminar on such issues. That would be a wonderful idea. So I think we are running out of time. Having said that, now I would like to close my talk with a big thanks to the college for giving me an opportunity. And I really congratulate, I think it is Dr. Lata is the convener, right? If I'm right, Dr. Lata, for having such a wonderful international conference. And I wish you all the best.
Thank you so much, ma'am. Thank you so much.
For a successful international conference. And I think after this conference, you should be able to come up with some important new leads in drug discovery and development. Thank you so much. Thank you very much.
Thank you so much, ma'am, for your enlightening words. And we would certainly like to listen more from you. Moving forward, we have...
That is indeed a very good idea, ma'am. We will be having a conference on this also.
Yes, ma'am. Moving forward, we have with us Professor A.K. Prasad. We are grateful to Sir for accepting our request to inaugurate the conference today. Professor Prasad is presently the head of the Department of Chemistry and Dean of Faculty of Sciences at University of Delhi. Sir did his PhD from University of Delhi in 1990 in the area of synthesis of bioactive polyphenolic natural products. After spending about a decade as postdoctoral fellow and visiting scientist at the University of Southern Denmark, University of Copenhagen, Max Planck Institute for Molecular Physiology, Germany, La Sapienza University, Rome, University of Massachusetts, Lowell, US, Professor Prasad joined the department as the reader in September 2001 and subsequently became professor in June 2009. He is a member of several national and international professional societies and has published over 200 research papers in journals of international repute, along with 10 patents. His research interest lies in the areas of synthesis of sugar peg-based amphiphiles as novel carrier agents, modified nucleoside monomers for therapeutic oligonucleotide synthesis, biotransformations, natural product chemistry, and synthesis of bioactive heterocyclic compounds. He has handled several research projects, including bilateral projects funded by DRDO, DBT, DST, NMPB, and University of Delhi. Sir has also been the visiting professor at JIST Japan and SDU Denmark. He has been the recipient of ACCTI Excellence in Carbohydrate Research Award, ISCB Award for Excellence in Chemical Sciences, CRSI Young Scientist Award, and many more such accolades. Sir, we are honored to have you with us today. I now request Professor Prasad to deliver his inaugural address.
Thank you very much, Dr. Ritu. I hope I am audible.
Yes, sir.
So, yes, so Professor K.N. Saraswati, Chief Guest and Chairman of Maitreyi College. Madam, I agree with you that this college is a college of happening, because I have also witnessed a lot of happenings of this college during recent time. And, thanks goes to the principal, Professor Haritima Chopra, for arranging all these activities in the college. Particularly for this conference, I will like to thank all the member staff of chemistry department for making this conference to happen on a very important and current topic of trends in drug discovery and development. And also all the faculty members and students who are involved in organizing this, help in one or the other way in organizing this international conference. And all the speakers and resource person, I could see that from different parts of the country and the world, they have agreed to come for this conference and give lectures, for the interest of students and young faculty members and other researchers. And of course, dear participants, I could hear that about 600 participants has registered for this conference, and in hundreds they are already connected with online for this inaugural session. It is a matter of great pleasure, to be part of this inaugural function, of this 2 days international conference on recent trends in drug discovery and development. Particularly at this juncture of time, when the whole world is really, under COVID-19 pandemic since last almost 2 years. Although development in medicinal chemistry, medical biology, or medical sciences has really transformed our quality of life as well as life expectancy. There is always a need of continuous efforts in development of newer drug molecules to fight upcoming diseases. That's what we have seen. Even for this pandemic, which has covered almost the whole world, we do not have any medicine for this new virus, coronavirus, and the whole world is trying to come up with medicine. Of course, we have come up with very good vaccines for this, but then we have to also slowly come up with medicines, and therefore this continuous efforts by scientists from different areas is required, to come up with medicines for newer upcoming diseases. Life expectancy in India during 50s was only 45 years, which has now increased up to 69 years. But this is not enough, because if you see other countries, for example, in US, the total life expectancy is 78 years. In UK, it is 81. In Japan, it is close to 90. So, this increasing life expectancy is mostly due to the contribution of development of medical sciences. So our quality of life and life expectancy both has become better and better in days because of the development in medical science. There is an incremental development in every areas of sciences every year. and SWAG in medical sciences or in its allied subjects. Chemistry and chemical biology, are also witnessing these incremental developments. And, simultaneously, its development is there, but simultaneously, we are also facing challenges too. What are these challenges? These challenges are to find out newer and better pharmaceutical molecules. There was a time when it was thought that, we have enough very good medicine for, bacterial or fungal infections. But slowly the community realized that all those best medicines, these bacteria and microorganisms has find out another ways. They have become resistant to these medicines. And then it was realized that no, we have to be a constant discovery of newer and better molecules, because it is a fight between different organisms. And if you want to get rid of these infections, you have to develop newer and newer drugs, because they also find the way to come out of these drugs by developing different pathways, metabolic pathways. If you see, the last year Nobel Prize in Chemistry was given to two scientists. One was Emmanuelle Carpentier and Jennifer Doudna and this Nobel Prize of last year was given, for developing a technology which is now known as CRISPR technology. And this technology is used to edit the gene. Just now we have heard from Professor Saraswati that diseases are reflection of genes and the environment. But gene is also one of the important part, because that's what we have just now heard, that in the same family, some of the persons are affected with this coronavirus, and some are completely free. There is no effect. So this is because of our different genetic makeup. So this tool, which was developed by these scientists who were awarded with the Nobel Prize last year, is a very important tool, because it gives you an opportunity, a technology to edit the gene. So you can correct the gene, or you can replace the disease gene with the new gene. So these kinds of things can be done. This is incremental development. Even you see this year Nobel Prize, it was announced, I think yesterday, or day before yesterday, to Benjamin and given to Benjamin List and David Macmillan. And they have got this Nobel Prize, for development of asymmetric organocatalysis. And this has also a great impact on pharmaceutical development, because you can synthesize stereochemically defined pharmaceutical molecules using, asymmetric organocatalysis. Here, the catalyst is not the metals, which was used earlier. Rather, it is simple organic compounds, which can be, which are biodegradable and can be taken care of. So therefore, every year there is incremental development in this pharmaceutical science, but this is not the end of the development. In fact, although they have the great bearing, but there is always a requirement of development of newer and better drugs to take care of these different kinds of upcoming diseases. And therefore, organization of this 2 days international conference on recent trends in drug discovery and development, will certainly provide an impetus to students and budding researchers working in different areas of medical sciences. Because I have seen the speakers, different speakers for this conference from different parts of India or from, from the globe. They have very different topics covering, covering the areas of drug discovery and development. And certainly it is going to help, the students and to all of us. It will provide an impetus for our future research. With this, I wish all the best to all the organizers of this international conference, particularly the principal of the college, who provide, the opportunity to teachers of the different departments, to organize conferences on different topics, and then particularly to the teachers of chemistry department, Dr. Lata, Dr. Ritu Gaba, and of course the teacher in charge, Dr. Rajni Johar, and all the other teachers of chemistry department for organizing, making this event possible, where a lot of students and young researchers can be benefited. So my all the best wishes for this 2 days deliberation of this conference, and thanks to all of you. Thank you very much. Thank you. Now I will, Ritu Gaba, please.
Yes, sir. Thank you so much, sir, for your inspiring words and wishes. Now, I would like to seek your permission, sir, to release the abstract book of the conference.
Sure, please go ahead.
Thank you so much, sir. Dr. Kiran, if you could please display.
It looks very nice. Good color combination.
Thank you, Dr. Kiran. We sincerely hope that this conference will provide a unique opportunity to learn about the latest trends and developments in the area today and become a stepping stone in the mission to create a valuable platform that enables knowledge sharing. We believe that this event will provide the best exposure to students, researchers, and teaching faculty and foster an environment conducive to deepen their understanding about the topic. I would like to thank you, everyone, for joining the inaugural session. We now begin with the first technical session of the conference, and Dr. Lata will take you further for the same. Over to you, Lata.
Thank you, Dr. Ritu. Ipsita ma'am, am I audible? Am I audible to all?
Yes, ma'am.
Yes, Lata.
Okay. Thank you. I welcome you all once again in technical session 1 of this conference. The session coordinator for this technical session is Dr. Ipsita Roy, one of our distinguished alumni. Dr. Ipsita Roy is an associate professor, Department of Biotechnology, NIPER, Mohali. She joined NIPER in 2005 and involved in teaching postgraduate and doctoral students along with many research activities. She did her BSc in chemistry from Maitri College, MSc in chemistry from IIT Delhi, and earned her PhD in applied biochemistry from IIT Delhi. She did her postdoctorate from Humboldt University of Bonn, Germany. She was certified as Radiation Safety Officer by Atomic Energy Regulatory Board from Bhabha Atomic Center. Dr. Ipsita Roy is the member of Institutional Biosafety Committee, NIPER, National Academy of Science, Allahabad. Her research interests include stabilization of proteins using small molecule and aptamers. Madam has more than 130 publications with high impact factor. Her H-index score is 38 and has been granted with 1 US patent. She has supervised a total of 11 PhD students and 85 MS Pharma students. She was felicitated with Distinguished Alumna Award from Maitreyi College in Feb 2018 by our Honorable Vice President Venkateswar Naidu. She has recently featured in top 2% global scientists in the list published by Stanford University in the field of biochemistry and also by the Life Science Organization as 365 Indian Women in STEM. I welcome you, ma'am. For this session, we have two invited speakers, Dr. Rajesh Bhardwaj from NIH USA, Professor Naseemul Huda from Department of Chemistry, Jamia Millia Islamia, University, Delhi, India. Now I request Dr. Rajni Johar to welcome and introduce our first invited speakers of this technical session. Over to you, Dr. Rajni.
Thank you, Lata. A very good morning, everyone. I, Dr. Rajni Johar, teacher in charge, Maitreyi College, welcome you all in today's first session of this international conference. It is my great pleasure to introduce our eminent speaker, Dr. Rajesh Bhardwaj, Senior Immunologist at National Institute of Environmental Health Sciences, North Carolina, USA. Today, Dr. Bhardwaj is going to deliver his lecture on the topic, Multidisciplinary Insights into Drug Screening and Validation for Calcium Channels Implicated in Health and Disease. Dr. Bhardwaj completed his BTech in Biotechnology from NC College of Engineering, Haryana in 2006. Later, he did his MTech in 2008 in Bioinformatics from University of Hyderabad, wherein he was also trained by Dr. Shekhar C. Mande, the current Director General of CSIR. Thereafter, he worked as a computational scientist with Dr. Nasreen at the National Institute of Nutrition, ICMR, Hyderabad. Dr. Bhardwaj obtained his PhD from Heidelberg University in Germany in 2013, wherein he worked on the physical coupling mechanisms between endoplasmic reticulum and plasma membrane. During his research career, he published a vast collaboration with more than 15 laboratories around the world and has published 18 high-impact research articles. He has more than 11 years of research experience on the drug screening and development for calcium channels implicated in health and disease, with the emphasis on understanding their mechanism of action. Recently, Sir has moved to USA, NIEHS, to study the role of calcium signaling in skin diseases. Dr. Rajesh, I am sure today we are going to get enriched by your knowledge and experiences. On the behalf of all the participants here today, I welcome Dr. Rajesh once again in this international conference. Thank you, and over to you, sir.
Hello, do you hear me?
Yes, sir.
Good morning, everybody. Just let me try to share my screen. Okay, thanks a lot for the very nice introduction and for giving me the opportunity to be actually the first speaker. So there is a lot of things that I would like to talk about. So as it was introduced that I'm going to talk about multidisciplinary insights into drug screening and validation of different calcium channels that are implicated in health and disease. So I'm working in the Signal Transduction Laboratory at one of the NIH institutes called National Institute of Environmental Health Sciences in North Carolina. And this is where North Carolina is in the US. And here you have the first in-flight signs on the car plates, because the first aircraft was actually flown here in North Carolina. This is the institute. We have a very nice lake just next to the institute. So I would like to begin with a very basic introduction that why calcium serves as a key signaling ion. So calcium has strong protein binding compared to the monovalent ions. It has high affinity to carboxylate oxygen and is the most frequent, which is the most frequent motif in your amino acids. It has a very flexible coordination chemistry. It has rapid binding kinetics. And as you can imagine, it is energetically favorable to use calcium as a second messenger because there is a large calcium gradient across the cellular membranes. So next, I'm going to talk about calcium homeostasis and why is high cytosolic calcium cytotoxic, that is deleterious to the cellular health. So high amounts of calcium can cause aggregation of proteins and nucleic acids. It can affect the integrity of lipid membranes. It can initiate the precipitation of phosphates. Therefore, the calcium in the cytosol, the intracellular calcium, is maintained to about 100 nanomolar concentration, which is nearly 10,000 to 20,000 times lower than the extracellular environment. And even the most primitive bacteria have some sort of calcium pumps that can remove calcium to the environment. So in 1880s, Sidney Ringer performed a very important experiment that led to a very important serendipitous discovery. So what Sidney Ringer did is he prepared his physiological saline, that is 0.75% salt. And back in the days, they were perfusing different organs, for example, frog heart. And he prepared his physiological saline with distilled water. And he observed the following, that the contractions of the heart, they would disappear after a while, right? So what happened is that in absence of Sidney Ringer, the technician in the lab, he prepared the physiological saline. And when Sidney Ringer performed the experiment, he observed that these oscillations actually lasted much longer. So that led us to led them to investigate that what could have been this reason that led to these sustained contractions and not this disappearance of these calcium oscillations. So it turned out when they investigated what is present in the water, then it turned out that the lab technician has used the tap water instead of the distilled water. And of course, later it was found that it contains calcium, and it is calcium that is important for sustenance of these contractions. So that really highlighted the importance of calcium, physiological relevance, and so on. And normally in several calcium binding proteins, you have different motifs. For example, this particular motif that is called an EF hand motif. So here you have these helices, E and helix F, that here have several negatively charged residues. And as you can imagine, that the negatively charged residues can undergo electrostatic interactions with calcium and can complex calcium. And these proteins having different motifs can thus have different affinities to calcium, depending on the positioning of these negatively charged residues and the number of these residues. So different proteins can have different binding affinities to calcium because of the presence of such domains. So here is basically to show you that this is a typical metazone cell. So what you see here is that the extracellular calcium is in millimolar range, about 1 to 2 millimolar. You see that the cytosolic calcium is maintained really low in nanomolar concentration, is indicated here, 50 to 100 nanomolar. Endoplasmic reticulum, as you can see here, is about nearly 400 micromolar calcium concentration. So endoplasmic reticulum is spread out through the cell, and it is considered as store of calcium. So nearly about 400 micromolar calcium is stored here. And then mitochondria also stores calcium in a micromolar range. So what happens, for example, if the cells undergo some sort of stimulus on a cell surface receptor, if an agonist binds, that leads to the generation of second messengers, which then activate here a calcium release channel called inositol triphosphate or ryanodine receptors in case of muscles. And so what happens is then basically the store of calcium. So the message here is that upon a stimulus, this calcium from the store is released into the intracellular environment, that is the cytoplasm. So when calcium is released from this organelle called endoplasmic reticulum, calcium is rapidly buffered by different proteins. For example, in this case here, calcium calmodulin is shown. So one calmodulin molecule, for example, can bind 4 calcium ions. So these kind of buffering proteins will rapidly buffer the released calcium into the cytosol. There are effector proteins which can bind to calcium, and this leads to activation of different physiological processes on time scales ranging from seconds to hours to days, different physiological processes. And these, you can think of these processes as calcium turn on processes, let's say. And then it is, as I said, it is important to clear this calcium out of the cells because you don't want to have a high cytosolic calcium concentration for a long duration. So what happens is, for example, you have this sodium calcium exchanger. So what sodium calcium exchanger does, it takes in 3 sodium and extrudes 1 calcium in return. So it's a exchanges 5,000 calcium ions per second. So it's really high capacity and therefore a low affinity to calcium. You have uniporter sitting in the mitochondrial membrane, and this uniporter is also uptaking calcium into the mitochondria. So mitochondria do function as buffers of calcium. And then again, on mitochondria, you have sodium calcium exchanger that does the same job. And then you have a pump here sitting in the endoplasmic reticulum membrane, SERCA. Basically, it needs ATP to function, as you can imagine, because you have to pump calcium against the gradient from a lower concentration to a higher concentration. So this leads to refilling of these stores, basically the endoplasmic reticulum. And then you have on the plasma membrane also sitting plasma membrane calcium ATPase, which is again a pump needs ATP. Again, you are moving calcium against the gradient. So this PMCA pump is extruding calcium out of the cells, and this coordinated action of different release channels or uniporta or exchanger or calcium pumps, this concerted action really ensures homeostasis of calcium and proper balance, and helps to maintain these concentrations in millimolar, nanomolar, and micromolar ranges. So, and if this is messed up, if you have imbalance of this calcium, it can, or high cytosolic calcium, it can trigger several cell death pathways as indicated here. So here is an example of different calcium channels, pumps, exchangers, or sensors that are present in mammalian cells. And what I'm going to focus is on the TRP channels here, as you can see, that are permeable to calcium, and store operated channels, ORI1, for example, here, that is also permeable to calcium. And please note that, as I indicated earlier, for example, mitochondria has this uniporta MCU. So different organelles in the cells can have different pumps or channels, release channels that can be regulated by calcium, or that are responsible for exchange and transport of calcium in and out. So TRPV6 channel is what I'm going to talk about. It's an epithelial channel, and this allows, so this allows the absorption of dietary calcium from our gut intestine. And what you can see here is basically the intestine cross-section and the lumen of the intestine. And on the intestinal epithelium, you have this channel present, the TRPV6 channel present on the apical side. And this channel is basically a tetramer. So you have 4 subunits of this channel that are in the plasma membrane, and that association or coming together of these 4 subunits leads to formation of a pore, which is very selective to calcium, and thus calcium can enter from outside into these cells. And then this calcium binding protein here, calbindin, is saturated with calcium, and this helps the calcium to accumulate at the basolateral side. And then the action of this pump sitting here, plasma membrane calcium ATPase, causes the release of this calcium towards the blood side. So as I said, TRPV6 is a member of TRP channel family. And I guess a lot of us must have just very recently realized the importance of TRP channels, because the 2021 Nobel Prize in Physiology that was given to David Julius and Ardem Pataputian for their work on TRPV1, which is a painful heat sensor that senses, for example, capsaicin, the red hot chili peppers, and piezo channels, the mechanosensitive channels. So is a very important class or family of ion channels. And specifically TRPV6 that we are talking about here is a calcium selective channel, as I said. And to sum up, it captures the diet delivered calcium ions into the gut. So it is involved in the whole body calcium homeostasis, and it plays an important role in maternal fetal calcium transport as well. It is important for bone homeostasis, and it is a promising drug target for several different types of cancers. For example, breast cancer, ovarian cancer, prostate cancer, and pancreatic cancer. So here is a very detailed schematic of the TRPV6 protein. So what you see here is the transmembrane domains, and transmembrane domains, as you might understand, is the domain of the protein that passes through the cell membrane. So this is within the lipid bilayer. So it has 6 transmembrane domains. And as I said, these 4 such subunits of these TRPV6 proteins come together to form an ion channel. And here the pore of the channel where the calcium permeates through is in between the transmembrane domain 5 and transmembrane domain 6. So what you see here is the amino acid sequence of TRPV6 protein. And different ion channels can undergo different mutations that can either lead to a loss of function of that particular protein, or it can lead to a gain of function. So the function of the protein can either be enhanced or reduced. And they can be associated with different diseases. And such diseases that are caused by mutations in different channels are called channelopathies. So here highlighted are, so just to let you know that TRPV6 mutations, for example, are associated with pancreatitis or with skeletal abnormality. And here you can see in I'm not sure if it's pretty visible, but it's not very important to note, but the key here is that there are different residues within TRPV6, mutations of which derived from Japanese, German, or French families have been associated with pancreatitis, for example, and they are in different important domains of the protein that disrupt the functionality of the TRPV6 protein. So how do we basically record the activity of this channel? So what one can do is one can do, for example, calcium imaging, and FURA2 is a calcium indicator that is that gives a very accurate estimation of the intracellular calcium levels, let's say. So here what you see is this FURA2 indicator, and what you see here is a acetoxymethyl ester here. And what happens is when this FURA2 permeates into the cells, the cellular esterases remove this AM, the acetoxymethyl ester, and then the the free acid FURA2 can then form complex with calcium, and here what you see is the calcium bound form of FURA2 that is excited at 340 nanometer. So here you see the increasing concentration. So on the y-axis, you have the emission at 510 nanometer. So what you see is that at 340 nanometer, the bound calcium with increasing calcium concentrations leads to increase in the intensity of the fluorescence of FURA2. And the calcium unbound form of FURA2, it gets excited at 380 nanometer. So here you have the zero calcium towards increasing calcium. So the excitation is higher at, or the fluorescence intensity is higher at a lower calcium at 380 nanometer. The emitted light, as you noted here, is measured at 510 nanometer. The fluorescence intensity so increases at 340 nanometer with increasing calcium concentration, as I indicated, and decreases at 380 nanometer for the unbound form. And what you do then is you take a ratio of 340 to 380 that indicates the cytoplasmic calcium levels. So this is how you can do calcium imaging and estimate the calcium levels in the cytoplasm. So what you see here is the activity of the TRPV6 channel. So the cells, for example, are sitting here in, these are HEK, human embryonic kidney 293 cells. So they are sitting in nominally calcium-free medium. And you see here this the Fura-2 ratio that I just introduced to you. So you apply from a 0 calcium to 1 millimolar calcium, and you see this increase in calcium intensity. So what is happening is that the calcium from the extracellular medium is coming into the cells through TRPV6. And you can see that the untransfected cells that are not expressing the TRPV6 do not uptake this calcium. So when we apply a drug, in this case 2-APB, the structure is shown here with these 2 phenyl rings, it's a boronic compound, you can see that this inhibitor is able to inhibit the, or the 2-APB is able to inhibit the calcium influx through TRPV6 channel. Right, so this is one way that you can do this ratiometric calcium imaging to record the activity of the TRPV6 channel. So another high throughput way to do this, and this is a very regularly used equipment in pharma industry that does a lot of drug screening and so on. It's called FLIPR. It's fluorometric imaging plate reader. So as you can as you can guess that you are going to record fluorescence again here. And here you can use, of course, different commercially available calcium sensitive dyes. And in this case here, the excitation and emission are indicated for the calcium indicator calcium 5. You have the excitation about in the range of 470 to 495 nanometer and emission in range of 515 to 575 nanometer. And as soon as calcium is going inside the cells, or the calcium concentration changes, you will have the calcium indicator reacting to this calcium. And outside you have a kind of a masking dye, so you don't get any background signal because of the extracellular dye that is present here. So what you have here is the LED excitation and emission banks on this device. Maybe I can change to a laser pointer. So what you have here is the CCD camera, the LED excitation and emission banks. So what you can do here is you can put your 96 well plate or a 384 well plate, and you have a pipette head here, a robotic arm, and you can apply different drugs or substrates. You can keep your drug plate here, your substrate plate here. You can have at least 3 different administrations, and you can record all the 96 or 384 well plates at the same time, and you can do kinetics and basically, yeah, you can do drug screening using this FLIPR. So this here are the FLIPR recordings shown again for the TRPV6 channel. And what was done in this case is cadmium was used instead of calcium. And the reason is that cadmium, the dye is more sensitive to cadmium. And second is that we avoid endogenous secondary signals associated with calcium. For example, if I would, yeah, so if you remember in the previous slide, when we applied calcium, you might note that the calcium is basically extruded from the cell, or the TRPV6 channel also has a mechanism called calcium dependent inactivation. So if you apply calcium, what happens is that this you have this declining phase of the calcium entry because of the endogenous different molecules that are activated to take care of the calcium extrusion or inactivation of TRPV6. And of course, here, if you use cadmium, you avoid all these secondary processes that happen with calcium and will not happen with cadmium. That's why you have this type of curve here with cadmium. And here, when no cadmium is applied, of course, there is no entry. And this is just a control here. So when TRPV6 is not expressed, you see that the difference of the cadmium entry between the control cells and the TRPV6 cells is really remarkably high. So you can do this kind of high throughput measurements of the TRPV6 activity. And as you can imagine, you can measure different mutants, you can apply different drugs, and so on. So there are a lot of options that you can choose to do in this case. So what was done here in this study is a 3D ligand-based virtual screening method for identifying TRPV6 inhibitors. As you've learned, that it's a very important drug target because of its involvement in pancreatitis or in different types of cancers. So what you see here are different weak TRPV6 inhibitors that were known in the literature. And you can see that the IC50 is really terrible, 90 micromolar, 200 micromolar, with only this tamoxifen having about 8 micromolar IC50. So what was done here is that a scoring system was set up. So basically what you see here is the atom pair proximity score. So you are basically, what you're doing is you're running a similarity search. So a seed molecule structure, what you see here, and for example, your query structure. And what you see here is the atom pair proximity score. So this, or so-called the ligand overlap score. And what this score does is it quantifies the spatial overlap between these two molecules that you see, for example, here in above in orange and green. And the spatial overlap is quantified as a weighted sum of these atom pair proximity scores. And so it's basically independent of the shape similarity between two molecules, but it's kind of relies on this proximity scoring system. And then there was this atom category extended ligand overlap score. So what happens here, it combines the LOS score we just spoke about of the hydrogen bond donor, hydrogen bond acceptor, and hydrophobic atoms, and some complicated formula to calculate this extended ligand overlapping score. And based on that, what you see here is the alignment of these, the seed molecule and the query molecule. And this XLOS value optimization is performed by several iterative translations and rotations. And you can see here along these three principal molecular axes. And so to cut the long story short, this ligand-based virtual screening, which relies on this atomic pair proximity scoring system, we identified, starting from about 50,000 molecules, we narrowed down and we found very nice hits. And we come down to identification of this drug that is called Cis22A here. And in the transform, so here what you see is the IC50 of the of Cis22A for inhibiting this channel TRPV6. And I've already introduced you how to measure the activity of the TRPV6 channel. So it was calculated the same way. And as you can note here, that the trans form of this molecule has a higher IC50 or is less potent, let's say. So here you can see again the cadmium entering through the TRPV6, and here you have the control. So these gray trays, for example, are the cells that do not express TRPV6 versus the black, which expresses TRPV6. And as you would note that 1 micromolar cis22A, for example, completely blocks, or it comes to similar to the untransfected cell. So it's a quite potent drug. And of course, when you're talking about a drug that you want to claim specific to particular ion channel or whatever your target is, it becomes very important to test its off-target effects. And as you can see, remember we just told you about the Nobel Prize. So this is the TRPV1 molecule, and you can see that this drug cis22A, it's basically not interfering so much with different TRP channels that we have tested here. Oh, this window keeps bothering me. So TRPV1, TRPV3, and different TRP channels and store operated calcium channels, which we're going to very shortly talk about them. So the off-target effects of these, at least on the TRP channels, at about 10 micromolar concentration, were not so bad. So and now the next question arose, or the next question we had is, how does cis22A actually inhibit the TRPV6 channel? So we at the laboratory of Professor Matthias Hediger, when I was in Switzerland, we joined hands with different teams and with chemists, our collaborators, Jean-Louis Raymond at the University of Bern, and electrophysiologists and physiologists in Austria, Professor Christoph Romanen, and here structural biologists in New York, Professor Alexander Sobolevsky, and actually Apu, who recently has a position in India at IIT, who did quite some work in this project as well. So basically what we wanted to do in this project is to solve the structure that how cis22A actually binds to the TRPV6 channel, where it binds, what is the modus operandi, and cryo-EM, X-ray crystallography, and so on. So just a very simplistic schematic here. What was the message from this study that we did is, so what you see here is a representation, a tetramer of TRPV6 protein. And what happens is that when calcium comes into the cell, the calmodulin protein that I told you in my first initial slides, it can bind to calcium. It undergoes massive conformational changes upon binding to calcium. And calcium calmodulin actually controls the activity of different channels, a lot of ion channels, calcium regulation. And what happens is calcium calmodulin actually plugs this TRPV6 channel. A particular lysine residue is poking inside the pore and thus completely blocking the calcium entry through the channel. And that is exactly why you see this inactivation of the TRPV6 calcium current or calcium entry, because of the calcium calmodulin partly. And what we concluded in this study, that this drug cis22A inhibits the TRPV6 channel in a very similar manner like the endogenous inhibitor calcium calmodulin, and thus we thought that the side effects of this drug should be minimal. So this is a crystal structure of rat TRPV6 protein, and here you see a brominated derivative of cis22A. that is resolved with rat TRPV6. So you see that the drug is bound at 2 different positions. So this position E is, this are shown 4 different subunits of TRPV6 protein in different colors. And you can see that one drug is bound here in the pore, and one drug is bind in a site which we call the lipid binding site, where different other lipids are bound in the channel. So basically the drug actually replaces some of these lipids. And we focused, today what I'm going to highlight is how we validated one of these drug binding pockets here. So here it's just a zoom view of the different binding sites, and you can see here different amino acid residues are highlighted. And what I would like to highlight here in the pore here, this is the pore where the calcium permeates, right? So you have here, and again, this is the rat structure, so the numbers are a bit different from the human structure, but the key here is that this tryptophan residue, for example, and several other residues are in close proximity to this drug at the pore binding site. And now when you see here, this is the human counterpart and the information from the cryo-EM structure of TRPV6. So we also resolved different cryo-EM structures of cis22A bound to the channel. And you here see, for example, this residue isoleucine at 575 position, and you have this D580, and here what you see is on the y-axis is inhibition of cadmium entry by 0.25 micromolar cis22A, the drug that I introduced it to you. So what you see here is nearly 70% of the channel activity is inhibited by 0.25 micromolar cis22A. But when you have this mutation, when you mutagenized this isoleucine to alanine, or this D580 to N, you see that there is hardly any inhibition of this channel. So the channel becomes immune to cis22A mediated inhibition. And here shown are electrophysiology traces. So what you can do is you can record this movement of calcium ions as current in electrophysiology, and this is a negatively rectifying current. So what you see here is just another readout for calcium entry, let's say. So what you see here, the quantified data, the same thing, that the I575A and the D580 mutants, they have a immunity to the cis22A mediated block of the channel, right? And then you see that this W500, the tryptophan at 583 position, when this was mutagenized to a phenylalanine, which has here again a poor block by cis22A drug that you can note. So and then you can see that the IC50 actually shifted about 75 times after having this particular mutation. So it really became resistant to block by cis22A at concentrations where it would fully block the wild type channel. So like this, with the site-directed mutagenesis approach and different functional readouts, we could validate that this is indeed the drug binding site, because mutation of different residues that are involved in formation of this binding pocket, the drug now fails to block this channel. So what we did in another study is we designed further advanced derivatives of this drug cis22A in an attempt to make even more potent derivatives, more soluble derivatives of this drug, more biostable derivatives of this drug. And one such way to assess the stability is what you do is you put these drugs in microsomes, liver microsomes, which have different enzymes. And what we did is we did an optimization based on this natural product capsaicin that is present in red hot chili peppers, again, an activator of TRPV1 channel. And what we did here is we borrowed different parts of this capsaicin moiety, and to cut the long story short, finally, after a screening of hundreds of compounds, what we came up is with this drug that we call 3OG, an advanced derivative of cis22A. And what is important to note here is that you see that the IC50 from 320 nanomolar changed or improved to 83 nanomolar. The other homologous ion channel TRPV5, which is very similar to TRPV6, but is more relevant for kidney. And that so the TRPV, there is some activity on the TRPV5 channel as well. And then the off-target effects. So here is an off-target effect profile. So in blue, you see the previous drug that I talked about, cis22A. So you see that the blue has quite some off-target effects, because previously I showed you that on TRPV, on other TRP members, it didn't have quite some off-target effects. But now when you compare on different ion channels and receptors and so on, you see that in blue indicated cis22A and in red indicated 3OG, this advanced derivative has much lower off-target effects. And as I told you about the microsomal stability assays in these liver microsomes with a lot of enzymes that could metabolize this drug, you see that the drug still is immune to this microsomal digestion for more than an hour, whereas this is half gone in about 6 minutes. This is the cis22A original drug. So we think we have our hands on some of the advanced derivatives that might perform better than the original drug cis22A that was identified. So just to summarize, what I showed you in this first part of my talk is an introduction to calcium homeostasis. I showed you the relevance of the TRPV6 channel in health and disease. I showed you how to record the TRPV6 activity. I showed you the discovery of the drug cis22A, how computational methods can be used, and so on. I showed you about the mechanism of action of this drug cis22A, how different methods can be combined, and functional assays and structural biology. What I didn't show you is parts on molecular dynamic simulations, where we try to understand how stable is the cis22A that is bound to the TRPV6 channel in molecular dynamic simulations. A lot of in silico analysis can be done to further understand this mechanism of action. And if somebody could please remind me time, if I have still a few minutes, I would go into the second part. Otherwise, I can actually stop here if I don't have more time.
Dr. Bhardwaj, I think we are almost reaching time. So maybe another 5 minutes or so.
Okay, excellent. So I can quickly go through the second part then. So I want to show you all is that the endoplasmic reticulum in the cells is a very dynamic organelle, as you can see here in this movie. It's not how we imagine this. So it's highly mobile organelle. And what you see here is basically a endoplasmic reticulum retained protein. And what you see here is that endoplasmic reticulum can contact different membranes, different organelles, for example, mitochondria, Golgi, endosomes, plasma membrane. So endoplasmic reticulum is so mobile and it makes transient contacts with different organelles. And it is important to note that these membranes don't fuse. So they just, contacting. It can be a protein-protein contact. It can be a protein-lipid contact and so on. So moving on, what I want to show you is this channel called ORI1 that is on the plasma membrane. Unlike TRPV6, it is not a constitutively open channel. So it can be opened by another partner that sits in the endoplasmic reticulum membrane called STIM1, and physical association of these two leads to opening of this channel. Dr. Upendra will, I think, if I'm correct, tomorrow talk a lot more about STIM and ORI, so I rather keep it short. So what you see here is the structure of this ORI protein in the plasma membrane. It has 4 transmembrane domains. It is a hexamer, and TRPV6 you learned is a tetramer. Here the pore is formed by the transmembrane domain 1s. Here highlighted are several important residues in this pore. So basically, it is the key player of a process called store-operated calcium entry that I'm going to show you in the next slide. So what happens, as I showed you in the calcium homeostasis slide as well, that when calcium in the endoplasmic reticulum drops, it needs to be refilled. And here endoplasmic reticulum wants to send a message to the plasma membrane that we need more calcium to refill. And not just to refill, but also to raise the calcium in the cytoplasm to drive several other important pathways. And you can imagine that this pathway is very important in activation of immune cells, T cells, in gene expression and cell differentiation and so on. So what happens is that this protein STIM1 in its inactive conformation is in a locked conformation. And upon release of calcium from the ER, it undergoes massive conformational changes. It binds to these lipids at the plasma membrane and then physically couples both membranes so that the distance between both membranes is less than 10 nanometer. And this distance is so narrow that even ribosomes cannot sit in these tight opposed membranes. So then what happens is STIM1 undergoes further oligomerization and it interacts with this channel sitting on the plasma membrane ORI1. And this physical interaction leads to opening of this otherwise closed channel ORI1, right? So remember that TRPV6 that I told you about, it does not need an interaction partner to be opened, whereas ORI1 does need it. And here the calcium, in fact, actually a Nature paper where Matthias Hediger, my former boss, also collaborated with David Clapham, claimed that TRPV6 is the channel responsible for this calcium release activated calcium current. But later it was disproved and it was shown that how STIM and ORI are the relevant players of this and not TRPV6. So what happens is when calcium enters, when ORI1 is opened, calcium is entered into the cytoplasm, and this is then pumped back into the ER via the action of this pump SERCA. And what you can do in the lab is you can inhibit this channel, this pump by a drug called thepsigargin. And now you see that the stores cannot be refilled by calcium. This will cause passive depletion of the endoplasmic reticulum stores and will lead to activation of this pathway of store-operated calcium entry. And what we've done is, again, using some sort of calcium imaging techniques with Flipper that I showed you. What we do is the cells are sitting in no calcium. You apply thepsigargin. Now you're releasing calcium from the endoplasmic reticulum. That's why you're seeing this calcium transient. Then we're applying here either the vehicle or the drugs, different drugs, and then we are adding back calcium. And that's why we see this calcium entry, which is referred to as store-operated calcium entry, right? So what you see here is that different drugs that can block it or drugs that can actually increase this calcium entry. So what we did in this paper is we made a library of these 2-APB derivatives. You can see here again the structure of 2-APB. So very interestingly, the halogenated derivatives where we had fluorine, chlorine, bromine at the ortho, meta, or para positions led to a better block compared to 2-APB. And you can see here at lower concentrations, for example, at 10 micromolar concentration, you can see, for example, this ortho bromo 2-APB, for example, led to a potentiation, whereas 20 and 50 micromolar led to a block. And you can see that different derivatives here, that they behave actually compared to the original 2-APB better. So here is the 2-APB, right? So you have to compare it to this one. So you see that this 20 micromolar dose of 2-APB, which does about 50% block with the parent drug, you have several halogenated drugs that perform much better than the original parent drug. So we think that some of these halogenated drugs have quite some potential, more potent than the original 2-APB derivative. We have yet to assess their off-target effects, of course. And this brings me to basically another potent drug that we would like to highlight is GSK 7975A. It's a drug from GlaxoSmithKline. So I remember I talked about the TRPV6 mutations that are disease associated. Similarly, in ORI1 channel, you can have different mutations, or different mutations are actually known gain-of-function mutations. Again, I think Dr. Upendra will talk to us more about this. But here, what I wanted to highlight, that these mutations lead to gain of function. So the protein is now active without the requirement of STIM1. So we've shown in this study that GSK 7975A can inhibit the activation of these channels. And here you can see the dose response curves, that at a sub-micromolar IC50, all these open channel mutants can be inhibited by this drug GSK 7975A in these patients, in these ORI channel mutants associated with tubular aggregate myopathy, which is kind of a muscle disorder. So to sum up, what I've shown you in the second part is that how endoplasmic reticulum contacts with different organelles for a number of reasons, for lipid transfer, for calcium signaling. I've introduced you to the concept of store-operated calcium entry. I've shown you some advanced 2-APB derivatives that are more potent than 2-APB as inhibitors of store-operated calcium entry. And I've shown you that the GSK 7975A, this drug from GlaxoSmithKline, is able to inhibit the tubular aggregate myopathy, a muscle disorder associated ORI1 mutant, so might be a potential drug there. And that brings me to the end of it, and I'd be happy to answer any questions that you may have.
Thank you, Dr. Bhardwaj. That was an excellent talk. And I do not think the organizing committee could have planned it that way, but having the Nobel Prize on a member of the same family of proteins this year, and then your talk as the first talk, is a very auspicious beginning for this conference, I think.
I can see there are, thanks a lot.
We have a number of questions here. So I do not think the time will be there for asking all of them. So the organizing committee has promised that they will email all the questions to you, and as per your time, but I'll go through at least a couple of them.
Right.
So we have a question from Prachi. So she asks, is there any specific hormone which is important for calcium homeostasis?
There are a number of Yeah, there are a number of hormones that are parathyroid hormone, for example. And actually, I would think that different hormone secretion and so on is under the control of calcium signaling, different transcription activation. It's a lot of things on a downstream control of calcium signaling. So there is, yes, the answer, short answer is yes.
Okay. Thank you. So the second, actually, the questions are a mix of calcium homeostasis as well as the drug discovery process in general. So another question that Uma has asked is, how is the calcium channel important for drug screening?
Well, there are, so calcium, number of calcium channels, they've been highlighted in different diseases, right? So it's very highly relevant, and cancer, especially that the cells, they undergo, because of hijacking the calcium signaling pathways, they gain resistance over apoptosis, they gain drug resistance and so on. And if you can identify a particular channel, be it calcium channel and whatsoever, and if you can identify a target that is responsible for that, it is a very very important drug target. And these are specified actually as oncochannels, and TRPV6 has been classified as an oncochannel because of its relevance in different cancers that I've highlighted. So a number of diseases, osteoarthritis, and you name it. So very
right. So I think that also partially answers what Prerna has asked about the multiplication of cancer cells and the calcium balance, and
right.
whether
So
calcium receptors can be targeted.
exactly. So the hallmarks of cancer, calcium signaling dyshomeostasis, I guess some people have talked about adding it as a hallmark of cancer.
Right. So Dr. Bhardwaj, we are actually running short of time. As you can see, there are a number of questions in the comments box, so which shows the interest which people have in this area, and your discussion in particular, and very good pieces of publication. So congratulations for that.
Thank you.
So
yeah,
the organizing committee will email you the questions, and if you can reply to them, then the answers will be forwarded to the participants.
Excellent.
So,
Yeah.
Thank you. Thank you so much for your time.
Thanks a lot. Thanks a lot.
Thank you so much.
It's a wonderful event, and I'm looking forward to
Yes, I agree.
Thank you.
Thank you so much. So we can move over to the next talk, I suppose.
Yes, ma'am.
Yeah.
So I can stop the.
Yes. If you can stop.
But I can maybe leave and rejoin then.
Yeah, that is also good. We would like to have you for the other sessions as well.
Thank you. See you.
Now I request to Dr. Ankita Chaudhary to welcome and introduce our second speaker, Professor Naseemul Huda.
Thank you, Dr. Lata. A very good morning, everyone. I, Dr. Ankita Chaudhary, Assistant Professor, Mathrai College, University of Delhi, welcome you all once again to the International Conference on Recent Trends in Drug Discovery and Development. It's my pleasure to introduce our eminent speaker, Professor Naseemul Huda from Department of Chemistry, Jamia Millia Islamia, New Delhi, India. Today, Sir is going to enlighten us on topic integrating computational biology with small molecule synthesis towards the development of therapeutic agents. Professor Naseemul had completed his MSc in chemistry in 1994 from IIT Delhi and PhD in organic chemistry in year 2010 from Patna University, India. He has published more than 28 research papers with high impact factor in various journals of national and international repute. Sir has more than 24 years of teaching experience and has taught many undergraduates and postgraduate courses. He has supervised many PhD scholars and postdoc fellows. His research interest includes drug design and computational biology. He's a master of molecular dynamics simulations, molecular modeling, and molecular docking to further study the protein-drug interactions and small molecule disruption of protein-protein interactions. He has successfully completed many major research projects under JMI funded innovative research activities, UGC, as well as ICMR. Professor Hoda is also a member of Kishore Vaigyanik Protsahan Yojana interview board conducted by Indian Institute of Science, Bangalore, and supported by Department of Science and Technology. On behalf of Mathrai College, I welcome you, sir, once again in this international conference. Thank you, and over to you, sir.
Thank you. Thank you, Dr. Ankita, for nice introduction. And is this visible to all?
Yes, sir, it is visible.
First of all, I would like to thank Dr. Lata and Professor Haritima Chopra, Principal, and other organizing committee of Mathrai College. Thank you. Thanks a lot for the invitation. Here, my talk is actually, this lecture is focused on the undergrad students. Just here, I will be talking about, because drug design is an interdisciplinary endeavor. That is why I will focus on how the journey of drug design starts with the computational biology tool and how it is integrated with a small molecule synthesis. So is it visible to all now? No problem? Nothing?
Yes, it is visible.
Yeah. So when we talk about, actually, this is our building where my lab is situated in the ground floor from gate number 7, Jamia Millia Islamia. It is in South Delhi, and recently it is doing well in research and other activities. When we talk about drug design, it is an extremely interdisciplinary endeavor, works mostly at the interface of chemistry and biology, requires expertise in the wide range of disciplines like structural biology, computational biology, and organic synthesis. And there are other many fields that can be incorporated or integrated with this to develop new drugs. I will start with 2013 Nobel Prize, actually, which laid the foundation for the computational biology tools. Actually, this was awarded to Martin Karplus, Michael Levitt, and Warshall for the development of multi-scale model for complex chemical systems. Their work laid the foundation for the powerful programs that are used to understand and predict chemical processes and have applications in drug design, guest host chemistry, and protein folding. And most of the computational biology tools we are using in drug design is based on their discovery. In our talk, my endeavor with drug discovery focuses on structure-based drug design. When we talk about a structure, so a structure of the target protein is essential, and the target protein structure could be obtained through X-ray crystallography, NMR spectroscopy, and high resolution dynamic structure could be obtained through cryo-electron microscopy. These are the various techniques that led to the elucidation of structures of the target proteins. Why structure is important? So when we focus at the binding pocket, we need to have information about the residues present at the binding pocket, how the residues are oriented in space, and how the key residues interact with the inhibitors at the active site. For these informations, we need to have a structure of the enzyme as well as co-crystal structure is important for this. So this is actually when we talk about the development of a drug called Relenza, an antiviral drug. its target protein is neuraminidase. So neuraminidase pocket, this is neuraminidase pocket, binding pocket, and this is sialic acid binding to the neuraminidase pocket. And this enzyme is essential for the survival of influenza virus, and that is why its inhibitors are good antiviral molecules. Here you can see that how Relenza, an antiviral drug, binds at the binding pocket of Relenza. When we will compare its binding pattern with that of sialic acid, which is the substrate for this neuraminidase enzyme, we will come to know that its binding affinity is better than that of the sialic acid. How we can predict that? The magnitude of hydrogen bond interaction and other interactions are far more favorable in case of Relenza as compared to the sialic acid at the binding pocket. And that is why the information regarding the active site, residue, its orientation and its binding pattern with the enzyme is necessary. Now, how we computationally mimic all these biochemical systems. Then for that, there are various biological tools, computational biology tools we use. One of them is docking and scoring. There are other tools are molecular dynamic simulations, etc., etc. There are other tools, but just to save computational time, we generally focus in the beginning. We focus on this docking and scoring techniques, which is less time consuming and it gives a rough idea about the things. Later on, we use high level computational calculations to do other things. So when we talk about docking and scoring, it provides information about the binding affinity of the ligand with the target proteins. Computational methods that mimics the binding of a ligand to a protein actually predicts the pose of the molecules in the binding site. The binding affinity of a score represents the strength of binding. Here, I would like to how this docking and scoring works. So what we do is we find center of gravity of the active site, and then we bombard the molecule directly at the active site in several millions of poses and conformations. What happens is it finds best fit in terms of geometrical complementarity as well as charge complementarity. So when these two complementarity methods are met, then it gives us a molecule which could be further developed, which can be developed into a real drug molecule. So generally, these methods are used to predict for the preliminary prediction of an active molecule. Here, for docking purposes, generally AutoDock is a famous software, and we also rely on Paradox software. We feel that Paradox software is far more robust and give more accurate calculation than AutoDock software. And mostly it is geometrical fit as well as thermodynamic calculations, which give various scores to the ligands or inhibitors. What are the factors actually responsible for the protein ligand interaction? Here, I'm not talking about irreversible protein ligand complexes. We are talking about reversible protein ligand complex or non-covalent protein ligand complex. So pi stacking interaction is important, hydrogen bonding is important, then steric, then ligand flexibility also plays an important role. Then non-polar contacts are there, it also plays a role. And steric clashes are there. Here, when we talk about pi stacking interaction, when you will look at the double helical structure of DNA, you will come to know that two forces are important in the stabilization of double helical structure of DNA. First one is intermolecular hydrogen bonding, and second one is pi stacking interaction. Actually, when we are going for the development of any scientific methodology or any scientific hypothesis, we must look at nature. Nature, lot of unsolved things are hidden in nature, from where we can get some idea to develop molecules. Here, while starting my journey towards this, I will start with my initial work, which has enhanced my interest in drug design and development work. It is, we all know that it is a common household name in India. It is curcumin, and we know that curcumin is a beta diketone. So when we will apply the basic chemistry, we know that curcumin is a beta diketone, and beta diketones, if it is an acyclic beta diketones do not exist in diketone form, but it forms enol form, and it is enol form. It is stabilized by intramolecular hydrogen bonding. But when we did computational study regarding the deprotonation of curcumin at pH 7.4, we came to know that curcumin is not going to exist in neutral form, but it will exist in anionic form. And in anionic form, what is that? Excuse me. In anionic form, what happens is this these two carbonyl groups, when you will look here, so excuse me. So excuse me. Yeah, please, sorry. So here we can see that the two keto groups are in same side. When they will form in an ionic form, these two oxygen atoms are anti to one another. This was the important finding we obtained, and it led to the development of a molecule. This curcumin in ionic form, this is the three-dimensional structure, a geometrical energy minimized structure, and it binds well to the calcium calmodulin protein dependent kinase CMK4. It was a very good inhibitor of CMK4, where it is finding very good hydrogen bond with the key residues, and it actually helps us to look at the molecular level of the molecules, how an inhibitor interacts with the protein at molecular level. This is the structure where it is having very strong hydrogen bond with valine 121, and this was not happening with the neutral form, and it led to the journey towards the development of new molecules. Now, this was the beginning of our work. Later on, we switched to two kind of development of two kind of therapeutic agents. One was the development of anti-neurodegenerative agents, and in second approach, we are focusing on the development of anti-malarial agents. In anti-neurodegenerative agents, we are focusing on cholinergic hypothesis, where we have acetylcholinesterase enzyme, which hydrolyzes acetylcholine into acetate ion and choline. It is acetylcholine is a neurotransmitter, and acetylcholinesterase enzyme hydrolyzes it, and therefore it reduces the amount of acetylcholine in the brain. The treatment approach is, when we develop a molecule which is the inhibitor of acetylcholinesterase enzyme, will elevate the level of acetylcholine neurotransmitter in the brain, and hence it will improve the cognitive impairment. Another approach we are adopting is amyloid hypothesis. Here, either actually base inhibitor, beta secretase inhibitor, gamma secretase inhibitor could be developed, or A-beta disaggregating agent could be developed to enhance the neurodegenerative condition of the patient suffering from these diseases. When we look at the active site of this enzyme, acetylcholinesterase, the active site of acetylcholinesterase is responsible for hydrolyzing the acetylcholine, lies at the bottom of a 20 angstrom atomic Golgi. And what we know about it is it has two sites. This is the site where hydrolysis is taking place. This is the catalytic triad. And this is another site which is peripheral anionic site. This is catalytic anionic site where hydrolysis of acetylcholine is taking place. But this peripheral anionic site is also taking part in certain activities. First of all, it is acting as an anchor. First of all, acetylcholine comes and binds with the tryptophan 279 at peripheral anionic site, and then this tryptophan 279 directs the acetylcholine towards the active site for hydrolysis. This is the crystal structure reported by these people, which shows that this is acetylthiocholine. It is associated with tryptophan 279 through pi-cation interaction, and later on it moves towards the tryptophan 284 present in the vicinity of catalytic anionic site. Now this is the crystal structure of acetylcholinesterase with the mainstay drug of anti-Alzheimer agent donepezil, where we can see that this tryptophan 84 at catalytic anionic site is having pi stacking interaction, and tryptophan 279 is also having pi stacking interaction with another aromatic bicyclic system. As we know that tryptophan is also having indole ring, which is also an aromatic bicyclic system. These two pi stacking interactions were really important for us, and by keeping this idea into mind, we developed something. Excuse me. We developed this molecule having comparable IC50 value with that of donepezil, and our molecule is also interacting with tryptophan 284 and tryptophan 279 at pass. So in that way, it is behaving as a dual binder to the molecule. The idea was we must have a chain length of the molecule such that it is covering a distance of more than 20 angstrom. It is binding simultaneously with catalytic anionic site and peripheral anionic site. And this work was actually performed by Dr. Jitender, who is presently an assistant professor in our lab. Then this work, based on the similar concept, we developed another molecule, and this kind of molecule where you can see that triazolopyrimidine ring is connected with triazine molecule through a piperazine linker. And just to cover the length, we also introduced something else. As we know that trifluoromethyl group is becoming quite fashionable in drug design endeavors because of its stability and the compact electron density fluorocompounds is having. The compact electron density of fluorocompounds in its stability towards hydrolysis and some other factors which helps fluorocompounds to be a better therapeutic agents. So here we used this one and it worked well for us. Here this work was performed in our lab by Dr. Mudassir and Dr. Ehtesham. Now, both of them actually, Mudassir is in Norway doing postdoctoral fellowship, and Ehtesham recently finished his postdoctoral fellowship from China, Wuhan, near to very maybe 100 kilometers from Wuhan. Then, excuse me. Something is happening. I don't know what. Then the first one was related to the development of acetylcholinesterase inhibitor. Now we wanted to incorporate certain properties in our molecule so that it can also disaggregate beta amyloids. As we know that beta amyloids are beta amyloids, maybe it is beta amyloid, maybe it is tau protein, or maybe it is alpha synuclein. All these proteins are misfolded proteins. And my idea was to develop something, some structural feature into the molecule which can interact with the beta amyloid sheets, aggregated molecules, and it can invade into it, intercalate into it, and sometime in some way, through some mechanism, it can disaggregate it. For that, previously, first of all, we are just sharing the TEM image of some of our molecule. It is the first one, panel A represents A beta 1 to 140, 1 to 42 at 0 second. B1 is after 3 days, that is aggregated A beta. And panel C represents its treatment with curcumin, which is supposed to be one of the best natural beta amyloid disaggregator. And D, E, and F, they are our compounds. And what we could see that our compounds are better beta amyloid disaggregator than that of the curcumin. This was another set of compound performed by Tarana Umar. Now she has also completed her PhD, and recently she joined Jubilant Organics. Later on, she wants to have her career in academics. That's why she has left the job there. It is based on it is a pyrazolopyrimidine derivatives. This is bispyrazolopyrimidine derivative, and doing well in their action. This is the two-dimensional structure which helps to design the molecules. And this is our hypothesis. The hypothesis actually, as we know that this is proline. Proline, we know that they are beta amyloid disruptors. We are beta chain disruptor. We also know that planar sheets actually they intercalate into the DNA structure, and we also know that this is piperazine ring is having a puckered structure. Puckered structure is not very thick, neither it is very thin. It is thicker than a planar bicyclosystem, but it is thinner than other three-dimensional molecules. So the combination of these molecules with proline we thought is going to be a good beta amyloid disruptor. And our results have shown these are few results which shows that these molecules are really very good beta amyloid disruptor. And our thesis is this because in the beginning we are not this piperazine ring, it is our three-dimensional structure is not present in the beginning, it is present in between. So what it is doing is the planar structure is intercalating between the sheets, and then the puckered structure is actually disrupting the sheets into a soluble structure. Similarly, this is some computational work. As I have been told to finish off early, so I will be moving some of, I will be just skipping some of my work. This is, as we know that alpha synuclein disaggregation on this C. elegans model. It was actually performed with association with Professor Dr. Amir Nazir at CDRI. And we got good results from here. This was also done by Mudassar Makbul. And this is actually pyrazolopyrimidine on pyrazolopyrimidine systems. Tarana Umar has worked, as well as Dr. Mudassir Makbul has also worked on this. These are few results. Now, this is another set of compounds which we did in association with Dr. Sharad Gupta at IIT Gandhinagar. And it was also associated with synucleopathy. And we were actually developing anti-disaggregating agents, or we were talking about disaggregating agents, but accidentally we got some interesting results here. What we got is our triazole-based compounds were showing very good affinity with aggregated molecules. On treatment with monomeric and aggregated alpha synucleins were found to exhibit a change in their fluorescence intensities. Few compounds showed 2 to 21-fold increase in fluorescence intensities with the aggregated alpha synuclein compared with the monomeric alpha synuclein. Shifts in lambda max was also observed. Red shift was observed in lambda max of several compounds. Selective fluorescent response of some of the compounds can make their use in cell imaging and can work as fluorescent probe. Same thing, two set of compounds and exciting results in a different direction. Then later on, we extended our work on anti-neurodegenerative agents. And here, the additional work was the blood-brain permeability. And our compounds are also showing very good blood-brain permeability. It is here. This we did in association with Stanford University. One of the research worker there, Mustafa Gavar, did this work for us. Our compounds had very high central nervous system penetration ability. They were comparable to that of donepezil, a mainstay drug for the treatment of Alzheimer's disease. And here we actually worked upon the development of deoxyvascinosine derivatives. As we know that vascinosine is a quinazoline alkaloid. So deoxyvascinosine compounds we developed. As when you will look at the donepezil structure, you will come to know that these two structures are our developed structure and donepezil. They are superimposable on one another. When we discuss about superimposability of one thing over the other, we can say that they have very much geometrical similarity. And it was also performed by several computational tools. These two molecules are very much similar in geometry to one another. They have some difference in terms of their affinity with the enzyme. As we know that with the charge complementarity is not that much there, but the activity was comparable, and these compounds also shown good Todd disaggregating capability. This is the result. This recently we have published, and these compounds again had high blood-brain permeability. Now, my second project is associated with the development of phosphoethylamine methyltransferase inhibitor. This PFPMT is an essential enzyme for the survival of Plasmodium falciparum and other parasites, but this enzyme is absent in humans, and that is why PFPMT is an important target for the development of anti-malarial agents. This work we did in association with Dr. Pawan Malhotra at ICGEB. First of all, we would like to know that the active site of this enzyme consists of histidine and tyrosine, histidine 132 and tyrosine 19. They are in close proximity to each other. As we know that histidine is having an imidazole ring, which is a very strong neutral base. It deprotonates phenolic hydrogen of tyrosine. In turn, the phenoxide ion attacks at phosphoethanolamine and deprotonates it and make it more nucleophilic, and which do methyl transfer reaction. So the presence of histidine and tyrosine in close proximity is necessary for the action of this enzyme. What we did, we started with actually cyanuric chloride, a molecule. When I was starting my career at Jamia 12 years before, then I was short of money. So I was looking for cheap chemicals from where we can start my synthetic organic chemistry journey. So just I got the structure of cyanuric chloride, and it was such a nice molecule. When I looked at this molecule, I was just excited, and the reason for excitement was this is the best substrate to develop a molecule on the basis of a structure-based drug design. The molecule could be grown in 3 dimensions in a desired fashion. Whether we want to grow the molecule in X direction, or in Y direction, or in Z direction, we can grow the molecule according to our requirement. And we developed few molecules which were good PFPMT inhibitor, and these are the results. Actually, this is the docked structure where our best molecule is docked, and the distance between histidine and tyrosine is 2.2 angstrom. Later on, after certain time molecular dynamic simulation, the distance between these 2 residues were 7.3 angstrom. Here you can see that the distance initially it is 2.2 angstrom, and after more than 200 nanosecond simulation, the distance between them was 11.6 angstrom, and the molecule was laterally entering into the active site in between tyrosine and histidine 132. And this is interatomic distance with simulation, and these are all exciting results. And this is another set of compounds we developed for anti-malarial agents. This is quinoline based compounds, quinoline carboxylate based compounds. Here we used molecular hybridization approach for the development of anti-malarial agents. Actually, this was done by Dr. Anju. Now, recently, she is in South Africa. Sorry, not South Korea for postdoctoral fellowship. This is the main slide where we used this computational biology tool and our molecular hybridization approach to develop the molecule with very good binding affinity. And these are actually falciparum, Plasmodium falciparum, falciparum 2 inhibitor. Falciparum 2 is cysteine protease inhibitor. Cysteine protease, actually, for cysteine protease, we are working on 2 methods. One is the development of reversible inhibitor. These are reversible inhibitors. We are also working upon the development of irreversible inhibitor, that is covalent inhibitors. These are the results. This is the way. This is some, and this actually, I think I would like to stop here because and now at the end, a structure-based approach was adopted to obtain potential ACE inhibitors. New class of pyrazolopyrimidine containing misfolded protein disaggregators were identified. Diphenyltrazine may serve as a promising fluorescent probe for the detection of alpha-synuclein aggregates. Mechanism of action of the lead compound molecules were proposed from the simulation studies. And I would like to stop here. And I don't know if.
Thank you, Professor Hota. Thank you for a very good talk covering 2 very interesting and important topics as well. I also especially like that you talked of your topic, of course, but you also introduced the students to a number of avenues that are open to them, not only chemistry, but from chemistry, simulation studies, linking it to biology and everything. So thank you for that.
Thank you.
That's
Thank
an
you.
additional advantage that our participants had.
Thank you.
I also agree with you that nature is the best teacher, and
Yes,
there's a lot we can learn from nature. This is something which is very close to what we also do. So I really like the way you could work with molecules which are able to disaggregate preformed aggregates, because the disease is normally discovered when aggregation is almost over. So that is it's a very relevant topic. So
one thing, actually, I'm not trained in biochemistry or biological sciences. I have very little knowledge about it. But the way small molecules like amyloid beta is behaving is really interesting. And I would like to learn from you people about all these things. It's really interesting to know about all these things.
I mean, a lot of work has gone into inhibiting the aggregation process. So where you start with the monomeric protein, there are not very many molecules which can disaggregate an aggregate which has already formed. And that is what is required in the disease. So from that point of view, these are very interesting results, especially since they are able to cross the blood-brain barrier also.
Yes, ma'am. Yes.
So
Recently, we did this study in association with. Okay. Please, ma'am. One more. Excuse me. I have not shown the. Let me show the this actually acknowledgement. Professor B. Jairam, my mentor, and he has motivated me to rejoin the research work. Actually, I was doing my research work at IICT Hyderabad under the supervision of Dr. Yadav and the present director S. Chandrasekhar. Later on, I moved to Patna University, where we did not have any research facility. That's why I left it. When I joined Jamia, with the encouragement of Professor Jairam, I started my research work again. And then Dr. Pawan Malhotra from ICTV, Manisha Tiwari from ACBR, Amir Nazir from CDRI, Dr. Sharad Gupta from IIT Gandhinagar, and Philip Rosenthal from University of California for their help and collaboration. Thank you, ma'am.
Yeah. Professor Jairam has been my teacher also, so I know that he is a great inspiration. So
yes, ma'am, it's great inspiration.
So we have a couple of questions here.
Yes, ma'am.
So one is, what are the early signs of Alzheimer's disease? Prachi wants to know the early signs of Alzheimer's disease.
Early signs, it's difficult to say, but the formation of amyloid plaques and this topathy, these two things are not early sign, mid sign we can say.
Right.
Early sign, I won't be exactly, because hallmark of these diseases are amyloid beta plaques and neurofibrillary tangles. So these things are there. So we just here I would like to, because I don't know what to say here, but there are so many things going on. When we talk about, because the amyloid plaques are formed, before that the oligomers are formed. Oligomers, we cannot identify oligomers with that much certainty. So if we could, then it would be a good thing to find the early signs of this Alzheimer's disease.
Right.
What are other questions?
Yeah. So one question, I think purely chemistry question. Why are we considering nitrogen-containing heterocycles? Why not sulfur or oxygen?
Sulfur and oxygen. Actually, when Dr. Bhardwaj was presenting, I wanted to ask him a question regarding the involvement of iodine and bromine into the active molecules. There are certain, when we talk about halogen compounds, So in halogens, fluorines are very good substitutes. Chlorines are okay. Bromines and iodines, they are not good substituted molecules to exhibit exciting biological activities. The reasons are the larger size of bromine atom and the electron cloud is not that much compact in bromine. Therefore, it creates lot of clashes into the active site, lot of clashes. When we talk about nitrogen or sulfur, so sulfur does some physiological actions. As we know that now I am trying to incorporate sulfur through cysteine molecules in some of our antibacterial agents. We are talking about in association with AIIMS Delhi for the development of DNA gyrase inhibitors, where we are developing some quinozoline derivatives, where we are also incorporating cysteine. The incorporation of cysteine is just associated with sulfur atom. It does some exciting physiological actions. What are they? We need to understand this. Presently, I am in nascent stage of understanding of their physiological action, but we know that they do great things. When we talk about fluorine atom or oxygen atom or nitrogen atom, as we know that we learned from nature, we learned from the double helical structure and other biomolecular structures. So here, when we talk about nitrogen bases, nitrogenous bases, which do play an important role in forming different kind of hydrogen bonds, where it behaves as a hydrogen bond donor, as well as it behaves as a hydrogen bond acceptor. At the same time, it is true with oxygen. When you look at the properties of water, ammonia, hydrogen fluoride, you will come to know that water is a molecule which creates a balance. It has two hydrogen bond donor, it has two hydrogen bond acceptor, two hydrogen bond donor with the two hydrogen atom connected with electronegative oxygen atom, while it has two hydrogen bond acceptor with the two lone pairs connected with an electron rich, very strong electronegative oxygen atoms. So that is why these molecules are incorporated, and nitrogen is an essential part of thing. What I am also doing is just to incorporate nitrogenous bases, nitrogenous heterocycles, and we are also incorporating some homocyclic systems or non-heterocyclic systems and hybridizing together to come out with new molecules. Is my answer satisfactory or requires some explanation?
I think it's more of disease specific and then the properties of the atoms.
One more thing, ma'am, Ipsita ma'am, I would like to mention about, I don't know, I had an idea about capsaicin derived compounds Dr. Rajesh Bhardwaj was discussing.
Right.
When we look at capsaicin tail, we will come to know that at tail it has isoprene unit. It was having an isoprene unit.
Right.
If we can change the length of isoprene unit, it may give, because his work was inspired by capsaicin. If he can change the length of isoprene unit, keeping that isoprene unit at the end, or multiplying the isoprene unit with molecules of his interest, he may come out with some exciting results. I don't know. I was actually excited, and I am looking forward to synthesize some molecules having such structural features.
Yeah, I think the organizing committee must forward your suggestion to Dr. Bhardwaj.
Thank you, ma'am.
He has already got, he has already got very promising results. So maybe tweaking the molecule may get him more interesting results. So the other question was more about molecular dynamics and computational studies, how they are important for drug discovery, and whether undergraduate students where can they learn about computational
SCF Bio IIT Delhi is the best place to do this. And Professor Jairam encourages undergrad students to do such things. Even his idea is to develop a program which a high school students can run. And I'm sure that PARDOC can be run by, it is such an interesting and simple programs. A high school going students can run those softwares to design a molecule of interest.
Right. Right. So that is very heartening to know that students take something from here and which they can continue with. And that's what I said. Your talk covered not only science, but the avenues that are available to students later on. So thank you for that. So there's one specific question about this software that was used to check whether molecules are superimposable or not.
Yeah. Superimposability to it's actually what we do is for super actually one even Dr. Bhardwaj has shown one very good software has shown, and it was interesting to know about. Okay, it is also superimposing one over the other as well as it takes care of the charge.
Right.
I will suggest students to yeah, this is let me show. Yeah. It is the ligand overlap score loss.
Yes.
Loss that I think it is a better software to do such things. And I will also go into it because till now I have not used loss, and I will look into it. I will go through the literature and I will try to use it.
Okay. So I think that's very heartening for the organizing committee also to know that not only the audience but the speakers also get to know.
His lecture was quite interesting and quite inspiring.
That is great. So thank you, Professor Hoda. I have to formally also thank you. So very enlightening.
And you are also from IIT Delhi, also.
I'm also
from 1994 batch. I did my MSc there, and then I moved to ICT Hyderabad to pursue my research work. In between, I got a permanent position in Patna University. Then I moved there.
Right. Very good to meet you. So I think we can close the session here.
Yes.
Right. Thank you so much. Thank you.
So just I am leaving, and I will leave, and then I will join.
Yes, sure.
Thank you.
Yes, am I audible to all?
Yes.
Acha. Thank you, Dr. Rajesh and Professor Nasimul sir, for sharing your experience with us on drug discovery and its development. And I believe that our students definitely get enriched with the knowledge of various tools of bioinformatics. Thank you, Ipsita ma'am, for coordinating this session and sparing your valuable time from your busy schedule for this conference. Thank you, Dr. Ankita Chaudhary and Dr. Rajni Johar, ma'am, for organizing this session. Now, with this, we conclude the technical session 1. Now we will meet again for the next technical session 2 at 1 p.m. after the break. Thank you all once again. Shall I start, sir?
Oh, yes, please.
Shada ma'am, shall we start? Are you there? A very good afternoon, all of you. I welcome you all once again in technical session 2 of this conference. The session coordinator for this technical session is Dr. Shada Pasricha. The Dr. Shada Pasricha is an associate professor in the Department of Chemistry, Sri Venkateswara College, University of Delhi. She has more than 20 years of teaching experience at the undergraduate level. Madam did her PhD in synthetic organic chemistry in January 2000 from University of Delhi. Dr. Shada Pasricha is a member of Royal Chemistry Society and American Chemical Society. Her research interest area include natural product chemistry, green chemistry, medicinal chemistry with focus on heterocycles, synthesis, and application of catalyst in organic transformation. She has published many research articles with high impact in several reputed national and international journals. She has been extensively involved in writing, publishing, and reviewing the e-content for undergraduate courses. In her academic career, she has received many awards for fellowship, such as Doctoral Fellowship, CS, Distinguished Teachers Award in 2010 by the University of Delhi, Bharat Jyoti Award from Ministry of from MHRD in 2016. I welcome you, ma'am. And for this session, we have two invited speakers, Professor Diwan Singh Rawat sir from University of Delhi and Dr. Padam Shri Mudgal from Department of Biochemistry, Daulat Ram College, University of Delhi. Now, I request you, Mr. Kamal Sharma, to welcome and introduce our first invited speakers of this technical session 2. Over to you, Mr. Kamal.
Thank you, ma'am. Namaskar, sir. Good afternoon, everyone. I extend a very warm welcome to all of you in the second technical session of this 2 days international conference on recent trends in drug discovery and development. I, Kamal Sharma, Assistant Professor, Mathai College, University of Delhi, once again welcome you all in this second session. It is my great pleasure to introduce our speaker, Professor Diwan Singh Rawat, Department of Chemistry and Dean Examination, University of Delhi. Today, sir is going to deliver his talk on the topic, Ray of Hope for Parkinson Treatment, Excitement and Comment during its development. Sir completed his MSc in 1993 from Kumaon University, Uttarakhand, and was a university topper. In the year 1997, sir earned his PhD from CDRI, that is Central Drug Research Institute, now in organic chemistry. Professor Rawat did his postdoc fellow from Indiana University, Bloomington, USA, and Purdue University, West Lafayette, USA. Sir is a fellow of Royal Society of Chemistry, FRSC, and was honored with C. and postdoc fellows. Sir is a visiting professor in the Japan Advanced Institute of Science and Technology and is an executive member of Indian Society of Chemists and Biologists and the elected lifetime member of the National Academy of Sciences. Sir is an associate editor in RS Advances and Nature Scientific Reports and is also actively involved in developing teaching material and review of textbooks. During his research career, he has established a vast interdisciplinary collaboration network with many laboratories around the world and has published around more than 157 research papers in reputed national and international journals with high impact factor. Sir has successfully filed 7 patents, and one of his molecules has already cleared preclinical trials and is taken up by the industry for its development as a drug for the treatment of Parkinson's disease. Sir has also co-authored many books on these chapters in the areas such as medicinal chemistry and drug discovery. One of his authored books is forwarded by Nobel laureate Sir Professor Derek Barton, FIS. Professor Rawat has successfully completed 11 major research projects. Besides being a good researcher, Professor Rawat is a good administrator, and over 16 years, Sir has worked at various higher administrative positions in University of Delhi as Chief Election Officer and Treasurer, Delhi University Students Union, Coordinator MTech, University of Delhi, OSC Press and Head, Graphic Arts Center, University of Delhi, Warden and Provost, Jubilee Hall, University of Delhi, Dean Works, University of Delhi, Chairman, Governing Body, DDU College, University of Delhi. Currently, Sir works as Dean Examination, University of Delhi, and Chairman, Governing Body, KMC College, University of Delhi. Professor Rawat is a member of many national selection committees, expert funding agencies, Board of Higher Studies, advisory committees, and committee of courses, etc. He is also the recipient of several awards in his professional career, such as Pratap Bhaiya Smriti Samman and VC Pratik Chinn Samman Awards, Professor S.P. Hiremat Memorial Award by Indian Council of Chemists, Professor R.C. Shah Memorial Lecture Award, Indian Science Congress, Young Scientist Award by Indian Society of Chemists and Biologists, Professor D.P. Chakrabarty 60th Birth Anniversary Commemoration Award, and many more. Sir was recently felicitated Special Appreciation Award by the Vice Chancellor of University of Delhi. Professor Diwan Singh Rawat is a versatile speaker and has delivered more than 175 invited lectures in various national and international conferences. Professor Rawat is a very renowned personality in the field of drug discovery and development. Sir, we are very pleased to have you here with us today, and I'm sure that you will enlighten us more about the topic by knowledge and expertise in this field. On behalf of Maitreyi College and all the participants present here, I once again welcome you, sir, in this international conference. Thank you, and over to you, sir.
Yeah, thank you. Just let me share the slide first. So can you see the slide?
Yes, sir.
Yeah, am I audible?
Yes, sir.
Yeah. Thank you very much. Thank you, Lata and Hema, for pitching in me to this program today. As you know, nowadays, my most of the time is utilized in the administrative work. So I hardly get a time for anything. And that's the reason sometimes the people who invites me are in trouble actually, because they need something which I'm unable to give in time. So that causes a problem. Let me just tell you some story about the discovery of the molecule which we have developed for Parkinson. And before going to that, let me just tell you some story about the drug discovery program. Sorry. So if you look at the literature, when we talk about drug discovery, till the middle 19th century, for the treatment of any kind of diseases and alignment the human being had, people used to rely on the natural source, that's the plants or anything else like that. But if you look at the old literature, you will find that the oldest written evidence of utilizing the medicinal plants for the cure of human disease was found in the Sumerian clay slabs in Nagpur in India. That was about 5,000 years old, and I have given a picture of that slab here. What it tells you actually, that was the first documented evidence of utilizing the medicinal plants for the treatment of sickness of the human beings. It comprises about 12 recipes of drug preparation, referring about 250 various plants, and some of them were alkaloids such as poppy, henbane, and mandrake. So these were listed way back 5,000 years old, and that was found in Nagpur. And if you go to Indian Vedas, this is the page I have taken from Sushruta Samhita. What it says, it is about 10,000 to 500 BC old. It says the Sushruta Samhita is one of the foundational texts of Ayurveda or Indian traditional medicine. And the surviving text contains description of more than 1,100 illnesses and 7,000 medicinal plants, including its safety, efficacy, dosage, and benefits. So this was listed about 1,000 years old in the Indian Vedas. But when we come about the synthetic drug molecules, the first and the oldest molecule that was synthesized in the lab for the treatment of any kind of sickness is a chlorine. I have given the structure over here. It was discovered in 1869 actually, and it was considered to be a sedative hypotonic, and it is still used in some part of the world. But the first analgesic and antipyretic molecule were phenacetin and acetinolide, and they were the simple derivatives of anilines, and they were byproduct of the coal tar. And again, it was discovered about 1869 or so. But another beautiful example is the willow tree. So we teach to the students how the willow tree acts as an analgesic, and it was discovered in way back. If you look back in 300 BC, it is listed over there the use of willow tree for the treatment of pain and the temperature. But this willow tree, the active constituent was a salicylic acid, actually what we call salicylate, and it was bitter in taste and it irritates the gastric mucosa. Later on, the synthetic derivative of this compound was developed by Hoffman in 1897, though the first synthesis was reported in 1853 by Gilhard. But Gilhard died very young actually, so he could not revisit his own synthesis. So the practical synthesis what we utilize now for the synthesis of aspirin was developed by Hoffman in 1897, and we know the uses of aspirin, how much useful this molecule is. But the major advancement in the medicinal chemistry took place when the penicillin was discovered. And we all know the history of penicillin discovery. This is what we teach to our student in the classroom. But I will tell you some other story of penicillin, which we never teach to the students. There are other people who are involved. If you ask a general knowledge questions to the students, who discovered the penicillin, the answer is Alexander Fleming. Just realize, because nobody could have imagined or realized the importance of the drug molecule to have a drug molecule around. Nowadays, we are suffering about the pandemic. We have seen the pictures just in the March or so, this was second phase of COVID, the kind of situation havoc was there, because there is no drug for the treatment of COVID. So if you don't have a drug for the treatment of any disease, you can see what kind of fate of the human beings are. So these are some of the pictures I have given over here. These are the infection caused by simple bacterium way back in 1930s or 40s. There was no drug treatment available for the treatment of any of those infections. And these are the pictures. But this gentleman, as we teach to the students, it was an accidental discovery of the penicillin. Discovery was accidental by Alexander Fleming. But what we did actually, what we teach them, we teach the students that one day he left the petri dish open. When he came after a week's time, he saw some mold develop, and that mold has lies the bacterial cell wall. And he reported that it has an antibacterial potential. Yet he wrote that this mold is nothing but a penicillin. What was his contribution to this field actually? So what he did, he published this paper in British Journal of Experimental Pathology in 1929. Let me tell you, he did not pursue this work after publishing this paper. If you look at the original paper, he has mentioned clearly that it can have an antibacterial potential. He could not reproduce it, he could not isolate or give the structure of the compound. The structure was given by Edward Abraham in 1942. Then Hodgkin did the X-ray structure. They got the Nobel Prize. And synthesis was given by Sehen in 1957. So this is where his involvement in the penicillin discovery ended. Just one paper. In 1936-37, Harvard Florey read the paper of Alexander Fleming. He got fascinated towards this. He thought it can have a great academic potential. He has written that the medicinal potential of penicillin was just a bonus, because it's an academic interest that forced him to work on penicillin. So he and Jane started on it, and they treated the first person by penicillin. The name of the patient was Albert Alexander. He was a British scholar, and he got a facial infection due to streptococci. There was no drug available for the treatment. One of his eyes has to be removed. So they were working in a laboratory, and they went to the hospital. They offered that this is the molecule what they have. They can use it for the treatment because there was no clinical trial available at that time. Since the death of a patient was certain when you get the infection due to streptococci, so the hospital and the family people agreed, and he was treated by penicillin. Since the quantity of penicillin was not good enough, the disease relapsed. At the end, he died. At that particular point of time, because the quantity of penicillin was not good, the fourth person roped in, that is Norman Hatley. He was a biochemist. He developed a makeshift mold factory and increased the production of the penicillin. As a result, the death rate dropped to 1% from 18% during World War I. But when we teach to the students, the literature, we look at the literature, sole credit was given to Fleming. Fleming, Florey, and Jane jointly got the Nobel Prize for their work in 1945. Hatley was ignored. The reason may be, I'm not sure, because the Nobel Prize cannot be given more than 3 people in a group. That may be the reason. But literature also forgotten to him. His contribution to the discovery of penicillin was summarized by the Oxford professor Sir Henry Harris. What he said: without Fleming, no Chain or Florey. Without Florey, no Hartley. And without Hartley, no penicillin. Yet, he was not recognized for his work till next 45 years. These are the pictures of these great men who discovered penicillin. Hartley is not there. But Hartley got a unique distinction in 1990, after 45 years of the discovery of penicillin by Oxford University. They gave him honorary doctorate of medicine, and that was the first non-medical person in the 800-year history of Oxford. He got only 2 awards. 2 awards only for this discovery. So as a teacher, as a scientist, we should teach a correct literature to the student. We must go beyond the textbook, what it is written over there, because textbooks are also written people like us. We miss many important informations available in the literature. Let me just give you one more example of the drug discovery when we are talking to the drug discovery, how risky this program can be. Look at this. This was the molecule discovered in 1957 by a German company called Grunther. The name of the molecule is thalidomide. And this molecule was sold to 42 countries across the globe. And the use of this molecule was for the treatment of morning sickness of pregnant women. After 5-6 years, when data was collected, more than 10,000 babies died when this molecule was taken during the pregnancy time, and those survived had permanent disability like this. When the structure of molecule was reinvestigated, what is the problem, why this molecule has shown this kind of toxicity? The chemist realized that it has a chiral center. So one chiral center means 2 enantiomers. So the molecule was a resveratrol. Tetrahydrocarbon was discovered by Levell and Montafort in 1872. So the chirality, role of chirality in the biological system was not realized or appreciated till the discovery of this molecule. This molecule taught a lesson to everyone. No, you look back the literature first. So this is what it has happened. So the discovery, the development of organic chemistry at different levels, how helpful it was. Look at this. 1900, the average age at birth in India was about 23 years. Today, it is about 65 years. And the credit goes to this branch of science, what we call chemistry. This is why chemistry is very important. So we must teach to the students when we talk about the stereochemistry. The stereochemistry cannot be taught without the example you take of thalidomide. Then you explain why that happened. So a student will appreciate the subject. So because I will be going to talk about drug discovery program. So at the end, nobody should ask me the question whether I have developed a molecule or not, whether my molecule is in the market or not, because this is what the story is. It says, if you make 10,000 compounds, one compound goes to the clinical market as a drug. It takes about 16 to 17 years. And the total cost of drug development is this much. You can count the zeros. So to avoid these kind of questions at the end of the talk, it is always better to show this slide. So if you see this slide, understand this slide, so you will not ask that question to me. Go back again, nature. So this is the plant. This is Catharanthus rosas. इसको हम सदाबहार बोलते हैं. And आप लोगों ने ये plant बहुत सारी जगह देखा होगा. You know, this plant is so beautiful plant. सदाबहार का ये हमेशा खिलता है. And it has been a source of 70 alkaloids. 70 alkaloids have been isolated from this small plant. Out of which I have given 2 structures here. One is brindolin, this is the molecule. Another is cantharine, this is the molecule. And both of these molecules showed poor anti-cancer activity. The same plant produced another molecule when these 2 compounds are covalently linked over here by a covalent bond. And this molecule, where you have brindolin and cantharine attached by a covalent bond, That molecule is called vincristine. And if you have a methyl group over here, that is called vinblastine. Vincristine and vinblastine are anti-cancer drugs approved by FDA for the treatment of leukemia in 1963. It is the nature who taught us. This molecule is not active. This molecule is not active. But when these two molecules are connected like this by a covalent bond, this becomes a drug molecule. So it means vincristine and vinblastine are hybrid of this molecule and this molecule. And nobody utilized this concept in drug discovery program till late 1990s. So there were two papers when I joined Naipur Mohali in 2002. Only 10 to 15 papers were there when we talk about conjugate. Some of the people says a conjugate, some of the people says the hybrid molecule. So we started thinking about those areas. So we thought to work on anti-malarial drugs. So this is the chloroquine, you all know, and this is the quinine isolated from a plant, and this is the artemisinin, isolated and discovered by Chinese chemist Yu Tutu, who got a Nobel Prize for its discovery in 2015. And what happened actually, these people reported if you make these kind of hybrid, this bridge comes from here, trioxane, and this comes from aminoquinine. And these compounds shows very good anti-malarial activity. So we designed our project based on that. So if you go look at the literature about aminoquinolines, aminoquinolines means chloroquine, and chloroquine used to be considered as a wonder drug for the treatment of malaria once upon a time. But later it has developed a resistance. The resistance mechanism is not well understood, but there are two broad hypotheses. One, if you give the chloroquine to a patient, it goes to the acidic food vacuole of the parasite, and dealkylation of N-diethyl group takes place, you get this molecule. And this molecule has low lipid solubility. So as a result, the bioavailability of the molecule decreases. So if you want to improve that, you can think to introduce such groups over here, which doesn't dealkylate very easily in the biological system. So the bioavailability can be number two, there is one enzyme called PFCRT in Plasmodium falciparum. What it does, it mutates the lysine to threonine. When that happens, this positive charge removes from it. So when the chloroquine goes in the acidic food vacuole of the parasite, you have a chloroquine here. So it gets a protonated, it stays, it shows the activity. But when the lysine is mutated to threonine, the positive charge goes off and you generate, the chloroquine efflux out. So you can inhibit that also. Based on that, we designed our molecule. Our design was take 4 aminoquinolines from here. So we have this one here. Another one is the other molecule, the anti-malarial molecule that was known in the literature is cycloguanine and premethamine. So if you look at these structures, so what we did, we chopped off this portion of chloroquine and chopped off this portion and hooked up those molecules by a covalent bond, something like this. And we thought, let's see what happens if you hook up 2 anti-malarial pharmacophores by a covalent bond and see what kind of biological response it shows. Let me just show you this. So if you have the 5-membered ring with 3 nitrogens, this is the activity. So this is the IC50 value of chloroquine. And if you look at this bar chart, the IC50 value of our compound is much higher than chloroquine. So it means this kind of hybrid has poor anti-malarial activity. Another hybrid was you have a triazine, then you have a pyrimidine here. So this is the activity chart. This is the chloroquine, and these are the IC50 value of some of the compounds. So the height of this bar decreases. It means these type of compounds have relatively better activity in comparison to the series what we did earlier. Next series was this. What was it now? Chloroquine, aminoquinoline, and trizine hooked up by a covalent bond, by a linker. What was the activity? This is the chloroquine IC50. And look at our compound's activity, better than chloro. So from this seminal work, we prove that this hybrid concept works well in the anti-malarial drug discovery. So as a medicinal chemist, you would think now, remove one of the CH of nitrogen of this trizine by CH, that will be your pyrimidine hybrid. So let's make those compounds. So these are the compounds what were made. You have 2 Cl in this, and this is more nucleophilic. Why it is? The student should read it, why the reaction doesn't go at this place. It's a good chemistry to understand it. So we got this intermediate. You have 2 Cls. So you have 2 possibilities of getting 2 kind of products here. We confirm the structure by X-ray crystallography and got the compound. So where we have are different groups, I will show you. So once you have these kind of compounds, we studied the anti-malarial activity of those compounds. So what was the difference now? Look at, this was the original design. Now this N is substituted by CH, that's it. Now, if you look at these 2 compounds, these are regioisomers. Regioisomers mean the connectivity of this group is at this carbon. In this case, it is in this carbon. This carbon is flanked by 2 nitrogen. In this case, 1 nitrogen nearby. Look at the anti-malarial activity. It is of similar kind. So the point of attachment of the aminoquinoline with pyrimidine doesn't make any difference in the anti-malarial activity of those compounds. So the major compound जो हमारा बना था उसको chemical modify किया. So we added these kind of carbocyclic compounds. Look at 0.21 to 0.026. The activity improves. Now from this we went to morpholine, then N-methylpiperazine, N-ethylpiperazine. Now activity is 7 nanomolar. So once you have a nanomolar activity, people will talk about the toxicity, number 1. Number 2, what about the in vivo efficacy of those compounds? So we did the toxicity studies, found that these compounds are non-toxic. Then when we did the in vivo activity of those compounds, chloroquine 100 milligram per kg body weight for 3 days, there was no treatment. Okay, but our compound 30 milligram per kg body weight for 3 days, 80%. So we prove that these compounds are active not only in vitro, but these are active in vivo as well. Once you have these kind of molecules with you in medicinal chemistry, we call it a lead optimization. So we did the lead optimization. So from where we started from this 2.45 micromolar IC50 where we reached 7 nanomolar. So the difference is this, in one case, in one of the series, this was variation was done at this place, or the variation was done at this place like this, or this was made a tertiary nitrogen over here, or the piperazine linker between these 2 pharmacophores, or piperazine linker with flexibility. So I will not discuss all those results. I will just concentrate these 2 examples and I will show you what happened. So this was the molecule, what we found at a 7 nanomolar activity. What we did in this case, we made the aminoquinoline and pyrimidine was hooked up through a linker that we call piperazine. So when we call a piperazine linker, it is a rigid linker because you will have a chair conformation. Okay. Not many flexibility there. And in another case, piperazine was kept, but flexibility was introduced there in terms of CH2, CH2. Were characterized by NMR. This is the NMR spectrum, filed the US Indian patent in 2017. What was the activity? So this is the original molecule. When you attach the piperazine in between the linker, in between the aminoquinoline and pyrimidine, the activity drops But when you introduce the flexibility over here, activity improves, and it is 50 times better active than earlier molecule that was 25 times better. What about in vivo activity? 33 milligram per kg body weight, 100%. Now you talk about the mechanism, how these molecules works. So we did heme binding assays. So what we found here, so when you titrate your compound with the heme, the absorption maximum of heme decreases it. So it means your compound is forming a complex with heme. So it drops like this. In the acidic food vacuole at pH 5.6 also, it drops. By job plot, what we confirm that the association of our compound with heme is 1:1. We did some molecular docking studies to see what is the role of this premethamine moiety there. What we found that it binds well in the native ligand of this dihydrofolate enzyme. So we don't have a experimental evidence for that, but we did some of those studies. Now, next design was based on the conceptual studies. So if you look at this molecule right here, this is called amodiaquine, and it is considered to be the better anti-malarial than chloroquine. But after some time, this molecule has to be taken back, withdrawn from the market. Now you can understand, if you remember the slide which I showed you, what it says, drug discovery program, 15 to 16 years, 17 years, number one, 10,000 compounds to one compound, $450 million. But if some industry spent this much of time and then withdraw back, you can understand the fate of that company. Why it was withdrawn? Let me just show you the basic fundamental chemistry. What happens when this molecule you take, it goes to the liver, it gets metabolized. When it metabolizes in the liver, this forms this kind of hydroquinone system. And that hydroquinone system is very toxic to the liver. And unfortunately, this was already known in the literature in the form of paracetamol. We all take paracetamol, right? So this is the paracetamol, and paracetamol also get metabolized in the liver in presence of P450 cytochrome enzyme, leading to the formation of this kind of benzoquinone. And this is toxic. That's why when somebody is suffering from a jaundice, should not dare to take paracetamol. If it does, then this can be very, very fatal, very, very fatal. Now, this was known because the people who were involved in the development of this were ignorant to this literature information. As a result, it was true. Now, as a chemist, if you want that this should not be formed in the liver, this should not be formed, what you will think? You will think two things. One, swipe the position of OH and this N-diethyl group. N-diethyl group ko yahan laga dijiye, OH ko niche le dijiye. It won't form. But we do not know whether the compound will be optically active or the biologically active or not. That we do. We can't predict. You have to do the biological studies. Another one is substitute this OH with a similar size of atom. This can be this. If you generate this compound, you won't get this. So toxicity issue will be solved. Whether the compound will be active or not, that's another question. People did that and found that this compound retains the activity edge. What we did this. So what we did actually in this case, we took this molecule, only changed what we made at this. And this was taken from our molecule, what we already had in our kitty. So this portion was hooked up with the fluoroamodiaquine molecule in a hope that this compound will show better activity, number one. Number two, less toxicity. Let me just show you how we made it. So this is again the synthetic route. I will not discuss it too much. So it is a simple chemistry. You get this, then after that you reduce it, CN2CH2 NH2, dichloropyrimidine, you get two regioisomers and you get the product. What is the difference between the amodiaquine, the drug molecule, and our molecule? This is the difference. That's it. In place of OH, we have F. In place of N-diethyl, we have pyrimidine ring O. Let me show you the activity pattern. Look at this. Chloroquine has 222 nanomolar activity for chloroquine resistant strain of Plasmodium falciparum. Our compound's activity is 5 nanomolar. So we had 4 compounds which has less than 10 nanomolar activity in this. What about the in vivo activity of those compounds? 33.3 milligram per kg body weight, 28 days, 100% cure. Stability, T-half of our compound is 16.7 minutes, and chloroquine, the amodiaquine has 5.4. This is what we proved. This is what we anticipated. Next design was, now, if you look at what is the difference here, this fluorobenzene was substituted by cyclohexyl group. Rest of the things kept same. This is the mass spectrum for the students. When you report the mass spectrum of a compound having halogens, you must report the M+2P. 553, 555. This 555 is because of Cl37 isotope. Remember that. So if you look at the antimalarial activity of this set of compound, so beautiful. Now the activity is 2.2 nanomolar, much better what we had earlier. $60 milligram for 7 days. After 60 days, all these mice survive, 100% cure. Not only that, their body weight increases. Not only cure, but it helped to regain their physical strength as well. At that particular point of time, actually, way back in 2012, we published this paper. When you read the literature, it gives you many insights. So I came across a molecule or the paper, which was published way back in 1987 by Sanders. It says, what this paper concludes it. This paper concludes the chloroquine or similar drugs could be used to retard the progression of Parkinson's disease in human patients in nature. Nobody pursued this. Our molecules are this much better than chloroquine. So we were interested to screen those compounds for Parkinson, but you need a good partner for them. When we published this compound, actually, in ACS Medicinal Chemistry later, one fine morning I received an email from Professor Kim from Harvard Medical School. He said that he has a high throughput screening system developed for Parkinson and he would like to screen our compounds. For that, he gave a genesis. What that genesis was? He screened 960 FDA approved drugs with the high throughput screening what he has developed. Found 3 hits. All these 3 hits has 4 aminoquinoline pharmacophore. That was talked way back in 1987. And we got interested to that. We started collaborating with Professor Kim. Before going to the Parkinson, let me just give you a brief idea about the Parkinson. Parkinson's disease, you know, it is a neurodegenerative disorder and estimated about 10 million people worldwide have the PD. The common symptoms of PD are tremor, rigidity, slowness of movement, and difficulty in walking. The motor symptom of PD results from the death of the neuron cell in the substantia nigra. This is the portion where the death of the cell takes place, which is located in the midbrain and it causes a deficiency of dopamine. So when this neuron dies, the concentration of the dopamine decreases. When the concentration of the dopamine decreases, you feel the Parkinson type of symptoms. Unfortunately, there is no drug available for the treatment of Parkinson till date. It is a dopamine, L-Dopa is given. It is for management only. But if the disease is progressed in the advanced stage, it doesn't work. So if you want to develop a drug for Parkinson, you should think about to activate some protein transcriptor factors of the receptors that are essential for the survival of dopamine neurons. The dopamine neurons are the neurons that helps in the synthesis of the dopamine. So if you stop the death of these dopamine neurons, you can have a drug molecule for the treatment of Parkinson. There is one target called NUR1, NURR1, NUR1. That NUR1 target actually protects the dopamine neuron from death, and it protects the neurodegenerative diseases also. But for a long time, excuse me, just a minute. Hello. Hello. So for a long time, it was found that the NUR1 is an orphan receptor. Orphan receptor means the receptor which does not have a binding pocket. The area where people felt that there can be a binding pocket was, surrounded by bulky amino acid, which is hydrophobic in nature. So it restricts of any of the molecule to fit in that side. But Professor Kim and his team confirmed that there is some unknown molecule which binds with the NUR1 and activates the NUR1 enzyme. When the activation of NUR1 enzyme takes place, it stops the death of dopamine neurons. So when the dopamine neurons death is stopped, the concentration of dopamine increases. And when that happens, the Parkinson's symptoms reduces. We started from that point. So I will not go to this, actually, you can read it. So we started screening our compounds. Initially, we sent about 120 compounds. If you look at this, this is the triazine and this is the pyrimidine. All the triazine-based compounds are totally inactive. These compounds are moderately active. Then it was funded by MJ Fox Foundation way back in 2014, the US, from the US side. We got a good funding on that. And we started making those compounds. Look at the EC50 value of chloroquine for this target, 50 micromolar. Look at our compound, 0.5 nanomolar. So we got many compounds which were low nanomolar active against this enzyme. So we had 15 hit molecules, and we proved that the T-half of one of the compound is similar to chloroquine. It has 75 better tissue penetration and 74% it get cleared better than chloroquine from the system. We did the biological studies were done by Professor Kim. I just want to show you some of the differences, what it is, maybe a little bit difficult for you people to understand. Let me just explain that. So what we were trying to find it out, that whether our compounds activates the NUR1 enzyme or not. For that, the assay system was developed where the luciferase was taken along with the receptor, and receptor's binding site was exposed to our compounds. So what was the result? Result was the chloroquine 100 micromolar activates the NUR1. Our compound 20 micromolar. So our compounds were 5 times better in activation to NUR1. So activation of NUR1 means what? The death of the neuron will decrease. Not only that, our compounds were better when it comes to the suppression of the immune system. It is much better than chloroquine. Chloroquine is this, our compound is this. And the protection of the cell, this is the vacuum. So if you treat this, give this with MPP, this is one of the molecule, and then treat that with chloroquine or treat with our compound. So this is the compound which causes the Parkinson. You induce the Parkinson, then treat with chloroquine. or treat with our compound. So you have more red spots, means our compound is much better than the chloro. This again shows the neuroprotection. I will skip this. And these are the in vivo activity models which were developed actually. In this case, what happens? This MPTP is nothing but a chemical compound that causes the Parkinson's disease. So animals were given this molecule first, got the Parkinson, then it was treated with L-DOPA, 50 milligram per kg body weight, chloroquine, 40 milligram per kg body weight, our compound 5 milligram per kg body weight. So when our compounds, these all three data points were collected and studied, what was found that the treatment is much faster and better when our compound was used for this purpose. Let me just show you the pictorial diagram, what happened. So we are talking about the brain. So this is the cerebrum part of the brain, and this is the substantia nigra, this one. So this is the healthy brain of a mouse. When this was infected with the compound called MPTP that causes the Parkinson, you can see the figure of the brain now, very dull. So it means the Parkinson disease has induced. When it is treated with L-DOPA, look at it, get little bit better, but not substantial. When it is treated with chloroquine, dose level is high, it regains. But if you talk about our compound, which is only 5 milligram, it goes close to the starting. So this is how we prove that this compound is really good, it's working. So 3 compounds were selected for this purpose, not for the development purpose. Not only that, the body weight of the compound, of the the animals also improves after the successful treatment. It has a good blood-brain barrier also. These compounds have very good pharmacokinetic parameters, important for the drug discovery. And this is the major breakthrough. We started from a molecule that was totally inactive. We got many molecules, battery of molecules at nanomolar level activity against the malaria parasite. Just a minute, please. Yeah, please call me after half an hour. Call me after half an hour. Okay, not only that, we got a molecule for clinical trials. And this is what the happiest moment. These 3 molecules are listed in the website of Nurwan Pharmaceuticals as a lead molecule for a development as anti-Parkinson drug. But the story doesn't end here. It doesn't give you the results like this. We always talk about good things to the audience. Look at this. When 2012 funding agencies funded one of the projects on malaria based on the concept what I discussed with you, they gave me unsolicited advice that the committee suggested that you should explore new ways from your routine work. That routine work was funded later on MJ Fox Foundation in the United States. And that routine work today is in the market, in the clinical trials. So it is the question mark on the wisdom of those so-called literate advisors and so on and so forth. The nightmare was not only with the funding agency, but my own university. Wanted to file a patent in 2012, 2013. This is what I had to write at that time to the registrar. I would like to pay from my pocket, please allow me to file the patent. I had 2 running projects at that time. Registrar take the 3rd option, which I gave, I will pay from my pocket. So I paid from my pocket. Technology transfer, what it came. We wanted to file an US patent. US patent filing cost about 10 lakhs rupees. My university will never pay that. So we have an MOU wherein it clearly mentioned all the the patent filing cost will be borne out by my collaborator. University has nothing to pay, but when royalty comes, it will be 50-50%. That paper signing took about 9 months. The most unfortunate part was when it came the technology transfer. The technology transfer was sent in February 2020, and it was signed in June 2021 when the new team came. So this is what it happens in our system, but if you are persistent, you can make it happen. What I discuss here today, it is because of the large number of students and the scientists who work with me day and night and made those hypotheses possible and brought some kind of laurel for the university as well. And these are the students, all of them are well settled at their respective places. And this is the group. This is not now the current one, because many of these people have completed their PhD and joined elsewhere. And the last one is this. This is what my genealogy is. This is who I am in academic field. I did my PhD with Dr. D.S. Bhapani from CRI Lucknow. He did PhD with Derek Barton. Barton did PhD with Hellborn. Hellborn with Hunch. Hunch with Wilkinson. Wilkinson with Hinge. Hinge with Rose. Rose with Berzelius. Berzelius with Abjous. Abjous with Bergman. And Bergman did with Benjamin Further in 1751 from Uppsala University. I did a postdoc with Richard Gibbs at Purdue. He did a postdoc PhD with Okamura, Okamura with Kurtz, Kurtz with Woodward, Woodward with Norris, Norris with Ramsden, Ramsden with Fitting, Fitting with Limchard. Limchard did PhD with Oler, the urea fame, and Oler did PhD with Berzelius and Gamlin, and Gamlin did PhD with Benjamin. Another postdoc was from Indiana University with Jeff Jaleski. I could trace this lineage up to 1637. With this, I end here, and thank you very much for your patience here. Yeah, over to you now.
Lata, can I start?
Yes, yes, please go ahead.
Thank you so much, sir, on behalf of Dr. Lata, the convener of this conference, Professor Ritma, the principal of Maitrey College, and on my personal behalf, I thank you for a very nice and lucid presentation. I think the sir has started with the history of drug discovery, the discovery of aspirin, and the importance of penicillin discovery. And he highlighted the disaster of thalidomide. He stressed on the importance of chirality to medicinal chemistry. From there, he has discussed about the antimalarials which have come up from his lab, the role of lead optimization and the beauty of molecular hybridization in drug discovery. And finally, sir has talked about how his compounds were able to activate the NER1 enzyme, which prevented the death of dopamine neurons and increased the dopamine concentration, helping in the treatment of Parkinson's. Thank you so much, sir, for a very nice and easily comprehensible presentation. There are a few questions that the students have put up. If you permit me, I will ask them one by one.
Yes, yes, please go ahead.
The first question is, sir, can you please explain the role of stereochemistry in drug discovery?
Okay. It's very simple actually. When we talk about the stereochemistry, we talk about the chirality, three-dimensional structure, right? So receptor is nothing, a protein that is made up of your amino acids, which has a three-dimensional structure. So if you have something like this is what we call a binding pocket. So you can fit something over here. So if it fits, it will show one kind of effect. But if your molecule doesn't fit with that binding pocket, fits somewhere else, it will show the toxicity. And this is what it was in case of the thalidomide. So it is a kind of, putting a hook in a right place. This is what it is.
Sir, there is another question. The student is asking, can we use molecular dynamics to check the viability of aminoquinoline for as a better drug candidate?
Yeah, actually, all those techniques can supplement to your studies. That's okay. But you have to prove it experimentally. There are many examples where, the molecular modeling or dynamics said that this molecule will have this kind of potential, and molecule was designed, and the experimental result was totally opposite. So it is always better, whatever you do the theoretical studies, it must be supplemented with the experimental result. Only then it has a potential. But based on that, you cannot unequivocally say, no, this is what it is, it will work. Had this been the success rate of drug discovery would have been much, much better.
Yes. Sir, the next question is, sir, what is your take on the report that the anti-malarial drug hydroxychloroquine can be used potentially for COVID-19 treatment?
Yeah, I was very interested about that because some of the things what I have not discussed, those molecules what I talked today had shown very good activity against dengue virus also. Okay, initially, if you remember at that time, the hydroxychloroquine was exported from India to many countries. But drug discovery program, the politics at the top level is really, really, you can't imagine because it's the money is involved. So yes, it's a good molecule to study with. It's a good molecule.
So one final question, the student wants to know if you're doing any work on COVID-19, sir.
No, because I didn't get any good biologist for that. No.
Thank you so much, sir. I think the young and the budding chemists who have registered for this workshop will be hugely inspired by your talk to take up drug discovery as a career in future. Thank you so much.
Thank you. Thank you. Thank you so much.
Thank you, sir. Am I audible? Achha. Thank you, sir, for sharing your knowledge with us on drug discovery and its development. And I'm sure that our students get motivated and enriched with the concept how the various heterocyclic molecules can be used as a better drug candidate. And sir, thank you for that, that you spared a valuable time from your busy hectic schedule. Thank you, sir. Now, I request to Dr. Pooja Baveja to welcome and introduce our second invited speaker, Dr. Padam Shri Mudgal. Dr. Pooja. Kiran, let her mic on, please.
She can do. She is
Okay.
co-host.
Okay.
Ma'am, please unmute yourself. Ma'am, please unmute yourself.
I think Dr. Pooja network is weak. Now I continue with this session. Very good afternoon, everyone. I, Dr. Lata Vodwal from Chemistry Department, welcome you all. And it's my great pleasure to introduce our eminent speaker, Dr. Padam Shri Mudgal, Associate Professor, Department of Biochemistry, Daulat Ram College, University of Delhi. Today, Dr. Mudgal is going to deliver her lecture on topic Zebrafish Model System, a tool for drug discovery. Dr. Mudgal graduated in Chemistry Honours from Miranda House, University of Delhi in 1982, pursued her master in biochemistry from the Institute of Medical Science, Chandigarh in year 1982 to 84. She earned her PhD degree in 1990 in animal biochemistry from Natural Drug Research Institute, Karnal, India. Her research interest lies in evaluating the therapeutic potential of traditional Indian medicine in various human diseases using zebra model system with purpose of developing drugs candidate. She has also gained expertise in using zebrafish model system in assessing the health impact of food additives and environmental pollutants. She has published many research papers with high impact factor in various international and national journals. Dr. Mudgal has a passion for teaching and has also co-authored a book titled Textbook in Membrane Biology, a Springer publication in 2017, which has become a very popular among undergraduate and postgraduate students. She is a recipient for many awards, include Best Innovative Idea in Teaching Excellent Award for Innovation by the University of Delhi in 2015. She also awarded with the Best Research Outcomes Award for Research Poster Display in 2016 at the Delhi University Convocation for the Innovation Project Zebrafish as a biosensor for assessing the Yamuna River water quality in Delhi NCT region. Recently, she was felicitated with Dr. Sarpalli Radhakrishnan Best Teacher Award in Biochemistry by the Center for Professional Advancement in 2021. Dr. Mudgal has expertise in zebrafish model system and biochemistry is present between us today. I am sure that we are going to get enriched with her knowledge and experiences. On behalf of all the participants here, I welcome you, ma'am, once again in this international conference. Thank you, and over to you, ma'am.
Thank you, Lata, for a wonderful introduction, and I thank you and the organizers for inviting me to deliver this talk. And I'll start my presentation. Yeah, you can see it, I hope. Yeah, I'm audible, Lata, and the screen is visible to you? Yeah. Okay.
Yes, ma'am.
Okay, thank you.
Yes,
so my topic today is Zebrafish Model System, a tool for drug discovery. Now, Dr. Professor Rawat has very illicitly described how difficult it is to discover a new drug and bring it to the market, and after clinical trials and reviews and all permissions. So drug development is a very lengthy, complex and costly process, requires minimum of 10 to 15 years, first spent on drug discovery, identifying the drugs and then their targets. Then you have the preclinical trials, then the human clinical trials. And of course, he has talked about very, very clearly about the politics of, permissions and regulations, and then how the drug comes into the market. So the whole story, the whole journey requires minimum of 10 to 15 years. Now, what we have seen in the COVID times that there is an urgent need to fast track drug development process to meet the current and future global health challenges. And many of you had, we know that many vaccines and drugs were brought into the market very fast, and many strategies were employed and are still employed, which are based on computational and AI-based strategies to fast track drug screening and discovery, to fast track preclinical screening, and to accelerate the approvals of the drugs which are passed through the clinical trials. Now, what I'll be focusing on today is fast-tracking preclinical screening. Now, for preclinical screening, a number of model systems have been used. The simplest, of course, is the in vitro models, where we use the bacterial or cell lines on which the drugs are tested. And then we have the in vivo models and many popular models. The mouse is the most popular, where most of the drugs have been tested. And of course, the Drosophila, but then Drosophila is far, far away in terms of similarity to humans. And mouse has its own disadvantages, ethical, very large life cycle and ethical limitations, and the number of mouse that can be used for testing. So one model, in vivo model, which is becoming very popular in recent times is zebrafish. And my talk today will be focusing on zebrafish as an in vivo preclinical model. Now, what makes this small fish with stripes so popular? Now, zebrafish is a small vertebrate endemic to India, small size, very low maintenance cost. External fertilization results in a collection of embryos of a high fecundity. That means each clutch, each pair of male and female can give us around 200 eggs. So if we have around 10 pairs, we can get around 1,000 to 2,000 eggs, which can give us a very good scientific validation and scientific data to rely on. The embryos are transparent, that you can see here on the left-hand side. The organs are visible, and you can actually phenotypically analyze and visualize the processes that are taking place. So that's one major advantage of zebrafish embryos. It's a very well-defined embryonic development endpoints. It's very defined at this point, at this stage, this will happen, and how it goes from a cleavage to a blastula phase to gastrula, and every phase is very well defined in zebrafish. Very interestingly, zebrafish, of course, the genome is sequenced, and it shows a 70% genetic homology to humans, and 84% disease-causing genes homology with humans. So very relatable. The results that we get in zebrafish are very relatable to human systems. Most of the organ systems are similar, and the signaling and the pathways are very similar to human. Another advantage, advantage of zebrafish is that most of the organ system develop by 2-3 days post-fertilization. So if you're performing a toxicity assay or doing a preclinical studies, within 5 days we have our results. We can observe the impact on the most of the organ system or at the molecular level. So it's a very good connect between an in vitro and an in vivo animal world. Apart from these advantages, zebrafish also provides additional advantages that we can genetically manipulate the embryos. So the embryos can be, and we can develop disease models. Like like Professor Rawat, we have Parkinson's models also developed in zebrafish, and so most of the disease models have been developed, and drugs can be tested by genetically manipulating, by functional genomics, genetically manipulating the embryos. And in our succeeding slides, I'll show you how that is done. So target validation and drug validation, drug safety and efficacy studies can easily be performed with good scientific validation and utilizing genetic tools. So it provides the simplicity of an in vitro system, but with the complexity of a whole organ system. So inter-organ relationships, signaling pathways can very, very easily be studied in zebrafish. For example, toxicity assay, drug toxicity assays can easily be performed in a high throughput zebrafish embryo toxicity assays by simply exposing the drugs to the embryos. So the embryos are collected post breeding the fish, the male and the female, and as soon as they are collected within they start dividing within 45 minutes, the embryos, then you can visualize it. And the treatment of the drugs is done usually at around 1,000 cell stage, that is around 3 hours post fertilization. And then they are observed up to 5 to 7 days for their toxicity studies, for survival rate, for hatching, and the impact on the different organs can be visualized. So phenotypic and morphological observation can give us, I'll show you the slide, the next slide will show us how we can identify the organ defects simply by observing phenotypic observation, and then we can go on for, do molecular studies and see what is the exact impact on the pathways, specific pathways. Now, these are the just example how toxicity assays are done. Now, this is an embryo, which is a normal embryo at 50 HPF. That means around 2 days, 50 hours post-fertilization, and the defects are visible on exposure to different chemicals. Now, you have heart edema, so that means there is an impact on the heart. There is a yolk edema, blood pooling. So these are observed as early as 2 days. And then around 3 to 4 days, now this is a normal larvae which is hatched. This is unhatched one. By 3 days they hatch, and you have body curvature defects which can be observed, and that can be related to the muscle defect or the motor defect, or you can correlate it by studying specific pathways. Now, very interestingly here, by day 7, if you see this is a normal embryo, the normal larvae which has got a developed swim bladder. Because the larvae swims, and then if you see this slide, this picture here, there is a very clear-cut darkening here. Now, this is the liver. So phenotypically, we can identify, or we can identify and see the hepatotoxicity induced by different drugs by day 7 simply by phenotypic observation. Now, this larvae also has not developed a swim bladder, which is again a developmental defect which has resulted. And there are some jaw deformities which are been observed, which are not present in the normal larvae. So simply by phenotypic observation, we can first identify the organs that are getting impacted, and what is the toxicity that is resulting by simple morphological studies. And then we can go on for which is the pathway or which system, which specific pathway is getting impacted. So zebrafish provides, you can do a screening, a high throughput screening of a large number of drugs, initial screening, if you start with 1,000, 10,000, or however drugs. So the first step of screening or toxicity assays can be performed in zebrafish on a large scale with minimum time within a 5-day period. And you can screen out the libraries, chemical libraries, and you can shortlist them to around, maybe 100 or 10 drugs, which can go further for, other in vivo higher models like a mouse model or other mammalian models, and before going in for clinical trials in humans. So apart from this, another thing which is very interesting, as I told you earlier, that zebrafish, we can develop or we can induce, we can develop disease models in zebrafish. Very easily, we can use specific chemicals again to induce diseases. For example, just to give you an example, for example, a fatty liver, we can just give in a diet rich in cholesterol or fructose, which can lead to a fatty liver disease or obesity model. And we have a lot of people in India are working on diabetes model. So diabetes can be induced by giving alloxan, or simple chemicals can be used to block particular pathway. So once you identify a particular pathway related to a disease, you can use specific chemicals to block those pathways and develop disease models. So disease models can either be developed by chemical tools or by genetic tools. So I think many disease models have been developed in zebrafish to study. Then once you develop a model, disease model, you can test your drugs that you have developed on these models and get the results that are expected. Now, many genetic tools have been used to develop disease models. For example, morpholinos have been used to knock out genes or to inhibit translation. Now, morpholinos are complementary oligonucleotides that are designed. You can design them complementary to the mRNA once you identify a gene, which is suppose, for example, a diabetes. Now, either an insulin gene or an insulin receptor. So you have a morpholino designed against which is complementary to the mRNA of insulin or insulin receptor, and these oligosaccharides actually are attached to the methylene morpholin ring, which replaces the sugars. So their shelf life is longer than the normal oligonucleotide. So these oligonucleotides complementary pair with the target mRNA and they block it. So that means they are inhibiting translation, so no protein is produced. So let's take example of diabetes. So no insulin is produced or insulin receptor is not expressed. So what you have is a morphin larval model. So this model can now serve as a disease model which you can test your drugs on. So mutants have also been developed by using other genetic tools like CRISPR-Cas. Now, Cas is the endonuclease which cuts DNA at a specific site. So once you endonuclease, and normal repair mechanism takes over, and this repair mechanism can either result in a deletion or a frameshift mutation or insertion, or maybe if you want to introduce you want to edit it and introduce a few sequences. So what you're getting is you're knocking out a gene of interest, and that means the gene is not being expressed, and what you get is a crispant or what you call it a mutant model. So that's how disease models have been developed. Before I go further, so which is the stage at which these are injected, these morpholinos are injected. Now the post-fertilization, as I told you that zebrafish undergoes external fertilization. So you collect the embryos after fertilization. It takes around 45 minutes before the first division will take place, right? So before that's a one cell stage. Now this is a one cell embryo. So it's in the first one cell embryo that microinjection is done. Microinjections is a very simple technique where you can introduce the morpholino. So it will be, as the cells are dividing, you will get mutants which are where the gene is blocked. So very easily reverse genetics results in developing mutants or morphins which serve as disease models. So just to give you example, number of disease modeling has been done with both chemical and genetic tools. And here you can see an example of diabetes where both by chemical use of alloxan, which is by intraperitoneal injection, that is in adults, or by giving glucose-rich or lipid-rich diet, diabetic models have been developed, or by mutation of insulin receptor. By developing a morphant or a crispant, we can use develop models which will serve as disease models on which drugs can be tested. So a number of diseases, I said thousands of disease models have been developed, like in Parkinson's, as Dr. Rawat mentioned with Parkinson's, even in infectious COVID model has been developed. So a number of infectious diseases models have been developed. So that's where the use of zebrafish as a disease model has been expanding tremendously over the years. So once you have these models, so you have developed these models, you can now, so by either by exposure or by genetic means, you have these models which are developed. Now you can test your drugs. Preclinical testing can be done. Once, of course, observation is phenotypic and morphologic observation, which is so simple to do just by simple observation under a simple stereo microscope. And then once you identify the defect, the organ defects, you can go in for molecular analysis or pathway analysis, cellular analysis, or behavioral analysis, and I will throw some light as I go along. Now, this was a case where I talked about how reverse genetics or genetic tools can be can result in disease model. And but then we can also use forward genetics have been used to generate transgenic reporter lines with fluorescent tags in zebrafish. So actually we can visualize the pathway, visualize the biological processes using fluorescent tags. For example, we can either use plasmids in which the promoter has been tagged with the fluorescent gene, which is a GFP or different colors. Many times two genes have been tagged with different colored fluorescent, either green or red. So you can introduce this plasmid again by microinjection in the one cell stage, or you can edit the gene with the CRISPR and introduce the fluorescent tag. And then in the first generation, what you get is a mosaic. Then you identify the heterozygous fish, crossbreed it to the first generation, and what you get is a homozygous. And I'll show you a few examples, beautiful examples, how these these fluorescent tags have really helped us in identifying the pathways and where the particular drugs can act, or what is the impact of the drugs on these pathways. So these tissue-specific reporters and gene editing technologies and drug activity can be studied at the single cell resolution using a fluorescent microscope. With the complexity of the whole animal, so actually you're observing a larva, a small larva live, so you're seeing the live processes and the impact of these processes live over an extended time scale. Just to show you an example, we are working on our lab, we have this transgenic lines where, the neutrophil have this enzyme myeloperoxidase, the gene is GFP labeled. Now, you know that the first response to injury, post-injury, is inflammation, that is migration of neutrophils towards the injury site. And you see these slides here, this is an uncut larvae, and then what you're seeing dots are the neutrophils which are labeled with GFP. Now, this if you can see it here, there is a cut here, at 0 hours post-injury. Here, there is a tail fin which has been cut. Now, within 1 hour, you see a migration of neutrophils. So this model serves a very good model for anti-inflammatory drugs if you want to test anti-inflammatory drugs, or if you're testing drugs which can, which can enhance or speeden up wound healing. Like wound healing is, as soon as you have injury, the process that starts is wound healing. So you want to study, and the first step in wound healing, of course, is inflammatory response, where you have a migration of neutrophils and macrophages. So you can tag even macrophages, and you can see their migration and how, then if you see after 3 hours, there is a resolution of the inflammation and the neutrophils start migrating back. So this transgenic line, this reporter line, is a very good model for studying anti-inflammatory drugs and processes which and studying inflammatory diseases, specifically like arthritis or diseases which involve inflammation, and it serves very well. Our lab, we are working on a few anti-inflammatory drugs, and serves a very good model for that. Another fluorescent tag model is GFP flea, GFP transgenic lines, where the flea promoter is tagged with GFP. Flea, this is expressed in the endothelial cells of the vascular blood vessels. Now, what you're seeing here is a 3-day-old larvae where all the blood vessels are visualized. You can easily see all the blood vessels. These are the blood vessels which are visualized. And what you see here is a sprouting of blood vessels and the process that's angiogenesis. Now, anti-tumor drugs, many of the anti-tumor drugs actually are targeting angiogenesis. So if you inhibit angiogenesis, naturally you're inhibiting the proliferating of blood vessels, and of course then succeedingly inhibiting the proliferation of tumor cells. So this provides a very good model. Just to give you an example how fluorescent reported lines have really opened up in the exciting area of visualizing processes, biological processes, and the impact of the drugs. So what you're seeing here is if you put in a drug, you can actually visualize and see whether it's having an impact on the sprouting and angiogenesis process. So coming to a different area, however, zebrafish has been extensively used. Zebrafish has got stripes. Now the stripes are made of 3 colors, which are brought to this stage due to the presence of 3 types of cells, which are melanocytes, which give its blue color, the iridophores, which gives its silver color, and the xanthophores, which gives its yellow color. Now these cells, are in a combination, are present in a combination, and result in giving the specific stripes to a fish. Now, why this is important is because these melanocytes have the pigment melanin. And melanin synthesis pathway is similar in zebrafish as in humans. So many of the human diseases, for example, albinism, vitiligo, skin cancers, can be, zebrafish can serve as a very good model here. And because the same pathway, melanin synthesis pathway, is working is present in zebrafish. Now, if you see this larvae here, there are, you see the darkening here, dots here, which are the melanocytes, and at least, 24 hours post-fertilization, the embryo start developing, or the melanocyte synthesis, melanin synthesis starts. The cell starts proliferating, and over time, you have more and more darkening of the larvae. Actually, you'll have to visualize the larvae, transparent larvae, you'll have to use an inhibitor of melanin synthesis. So if you see here, you see the drugs here, which are impacting the inhibition of the melanin synthesis. So a lot of labs, I think many labs in India are also working on vitiligo, and which is actually a very debilitating disease to a person who has it, and serves as a very good model associated with skin pigmentation and diseases associated with skin pigmentation. One area which has really excited us, and we are also working in this area and excited the world, is why zebrafish really gain into prominence, is regeneration. We have lost the capacity to regenerate many of our organs, but zebrafish regenerates its fins, its heart, its skin, even the retina, the photoreceptor cells, the retinal neurons post-injury. So serves as a beautiful model to study regeneration and of course, preceding regeneration, wound healing. So you in the embryo, very simplest model here, where you have a fin regeneration working, and here you see a heart. I'll spend a few minutes on regeneration, and a lot of work is going on in signaling pathways involved in why have we stopped regenerating? That's a big question. Now, where does the wound healing stop and regeneration begin? That's a very interesting question. Wound healing, most of our wounds, of course, heal. And that's how we stop at wound healing with the scar tissue formation. There is no scar tissue formation in zebrafish. It all starts with injury. Followed injury, you have the first, the step is the inflammatory phase, where you have a neutrophil migration and, migration of other cytokines, release of cytokines and process like that. And then you have a migration and, remodeling of the epidermis and regeneration of the lost damaged tissue, and then followed by proliferation. And then, of course, the maturation stage. Now, something between the proliferation state and the maturation state, something has been switched on in higher organisms, in primates and higher organisms, where we stop at the wounds healing stage. And we are not going into the regeneration state. So the question here, we are looking into the pathways which could, progress, how progress takes place from wound healing to regeneration, and how we can speeden up the process. Especially people with large wounds take a lot of time in healing. So we are working on certain drugs which can, really speeden up so the wound healing process and the regeneration process, if possible. And if it can be induced in humans, then there is no limit to what we can actually do. Now the advantages, these photographs have been taken by students in our lab. Now this is a photograph showing you the development of larval tail fin regeneration, right? So this is a normal fin. Now once you cut it post amputation, I think this is immediately, this is 1 day. This is a normal regeneration process. That means 1 day post amputation, you see here there is a blastema, this is the shrinkage happening, and then you have your progression, and then by 3 days you have a regeneration of the tail fin regenerated within 3 days. So you have such a fast process, such a fast model, where you can experiment and test your drugs, and you have a regeneration of a fin within 3 days. If you're working on an animal fish, on adult fish, then if you cut a tail fin, so post amputation the regeneration takes around 9 days, right? So you can study. Another interesting question is, why does what are the programs that direct the fish to take up the same shape? Now, what stops it to not further regenerate and progress from here and keep on regenerating? That means, what are the signaling pathways that define and regulate the shape that it takes and define the length that it takes? That means rarely it will go beyond, suppose this is around 5, maybe 1 millimeter. It won't go beyond 2 millimeter. Why? What stops it? So these are the pathways that really needs to be looked into, and zebrafish serves as a beautiful model to study, these processes. So So zebrafish is not just a fish, it's a hope. The zebrafish can heal its own heart. Now you have a heart, if you amputate the heart either by cryo amputation or different means, so it will regenerate the heart within 3 days post amputation. Now this is of course adult heart, right? So with adult heart regeneration now, so zebrafish can heal its own heart, maybe with some help, maybe we can heal ours too. So there'll be some years before, it won't be far that we know that we don't require a heart transplant, or we don't require, processes where heart surgeries are required. We be able to regenerate the human heart maybe in a few years. So that's the hope, right? So zebrafish, apart from this few of these models, another area that has really excited us is the neuropharmacology, and pharmacology where zebrafish serves as a beautiful model. Now, zebrafish being the brain is almost homologous to with the organ system is homologous to with the humans. So equivalent cellular and synaptic structures, function, the neurons almost similar to humans. It also exhibits complex behaviors like social behavior, anxiety, learning, memory, and defensiveness. And of course, the beautiful models, as I was telling you earlier, have been developed in zebrafish. Even the epileptic model, depression models, Parkinson's models, people are working on these disease models. So it responds to stress like in humans, and immediate endocrine response to stress. We are working on these anxiety stress related system, and it has a similar has a prevalence of an equivalent hypothalamic pituitary adrenal axis. They don't have an adrenal axis, but they have an HPI axis, which is hypothalamic pituitary internrenal axis, which serves a similar function as in humans. So a lot of similarity, and it's really gaining popularity as a neuropharmacology model. Just to give you a picture here and just to give you an idea. So a lot of behavioral essays have been developed in zebrafish. Very simple essays that can be performed in labs, and a lot of anxiety, for example, thigmostaxis. Now we know that when a child is stressed or is in distress, child goes and hugs his mother. Similarly, zebrafish also, larvae specifically, also try hugging to the wall when they are stressed. And I'll show you this video to give you an example. So the thigmostaxis essays can easily be performed. Light and dark preference. They prefer dark when they are stressed. So you can count the number of larvae which are in dark and light, and you can perform simple essays. Behavioral essays, anxiety or different impacts also impacts their movement, their motor movement, their locomotion. Many larvae, when they are for example, in anxiety, they freeze, they don't move, they're not dead, but they don't move, they freeze. So there is a locomotion derangement that can easily be observed. And as I'll show you as we go along with the slides, they also show learning and memory essays. And they show behavior. Social behavior essays can easily be done both with the larvae and with the adult zebrafish. And just to show you this video here, you can see how that this is a control where you have large number of larvae in the center, and not many are hugging here. Now you see here, these are stressed. They've been treated to some stress, I think to osmotic stress. So you will have more number of larvae hugging the walls. So you can easily take a video, a simple video using your mobile can be taken, and then you do a lab set, and you can easily do simple essays, behavioral essays, and count the number of larvae which are in the center and which are wall hugging. So a lot of I think many thousands of behavioral essays have been standardized to study behavior, zebrafish behavior, which you can easily correlate with humans. So now, of course, if you have the money, you can really buy these machines, which you can perform high throughput essays, locomotion essays. So you have these well plates, a 96 well plates. You can easily put them, and so you have your machines which can take do these essays for you, and analyze these essays for you. So a lot of labs are using these machines, behavioral essay machines, with locomotion, video trapping and analysis of the results. So high throughput assays to test large scale high throughput assays can be used to test psychosomatic and neuroactive drugs. And so you see, just to give you an example, how different locomotion behavior can be observed using these sophisticated instruments. Now here, what you see here is freezing. The larvae has not moved at all. Now here, what you see here is a water wall hugging behavior. Almost the larva is moving, around the walls here. And here you have a movement which is quite fast and all over the plate. So these locomotion behavior can easily be studied, and your psychosomatic and neuroactive drug and neuropharmacology has really taken a leap with the development of these sophisticated high throughput assays and instruments. So recently, I just looked at this paper where a study, what is the impact of COVID infection was studied, and they did a biochemical, physiological, and neurogenerative impact of SARS-CoV-2 infection was studied, and this was published in 2021. So they have done it in embryos. They infected the embryos and the larvae and in the adult, and they observed, it's a beautiful paper. So there's no end to what zebrafish can really do. You can make it an infective model. You can use it, develop a disease model, and then see the impact of. So this paper gives us a very good insight into what is the impact on neurodegenerative impact, physiological impact, and the biochemical impact of COVID infection. And even once you have a model ready, a COVID model ready, or a SARS infective model ready, you can test your drugs very easily. And another area that our lab is working on is using a zebrafish as a learning and memory model here. Very simple. It's so interesting that we were testing some, as Lata mentioned, that I was interested in food additives. So we were testing the impact of food colorants on learning and memory. So very simple assay. So what we introduced is we just introduced a straw, black straw in the well plate. Now, what was observed is what is known is actually when zebrafish larvae sees a new object for the first time, they view it with the left eye. So this is a left eye viewing. This is a left eye viewing. And and as it gets accumulated and gets known to the new one, when it is exposed next time, so it will observe it through right eye. Right eye. So known object is observed with the right eye, and the new object observation is through left eye. So if you expose it for a longer time and keep on exposing it, there will be a shift from a left eye to a right eye so that you can count. And one of the students did a work on it, and we observed that food colorants have an impact on learning and memory, because once you learn that it is a new object, that now you have learned about it, then it goes into the memory. So when you are exposed to it maybe 2 hours later or 1 hour later, you see what is the impact, whether the larvae is shifting to the right eye or not. So we saw that there is inhibition in learning as well as memory, and the impact of these colorants on can be correlated to what happens to humans and children who are really exposed to candies and drugs which have a high proportion of these colorants. So I conclude my talk here. So we know to conclude that new technologies and gene editing tools have really opened up the immense potential of zebrafish model in drug discovery. So I thank our principal. This is a zebrafish lab, and this is zebrafish habitat system where we house the zebrafish, which is auto controlled. And I really have to thank the principal, Daulat Ram College, for all support, our collaborator, Sanskriti Foundation, Delhi, and our financial supporters, Star College Project, DBT Government of India, Innovation Projects, DU, and Star Innovation Projects, Delhi University. So thank you all, and I'm open for questions. Thank you. Should I stop here? Yeah. Yes, Lata, over to you.
Rasicha ma'am, are you there?
Hello, thank you, Dr. Mudgal, for a very coherent and a very exquisite presentation. I think we've had two very productive sessions. First, Dr. Rawat, he emphasized on how science can be used to discover molecules. And now Madam Mudgal, she has explained how once you have a drug molecule in hand, how you can use those drug molecules to screen them for their preclinical potential. I think she started with the need to fast track drug screening and drug discovery, and how a zebrafish can be used as a model to fast-track preclinical screening. She emphasized why zebrafish is a viable preclinical model, the use because of its low cost, easy genetic manipulation, and high throughput screening potential. And then, she has explained how disease modeling can be done using zebrafish and how to test drugs, in a zebrafish model. She has explained the role of zebrafish model in skin pigmentation diseases, the role of zebrafish in regeneration studies, neuropharmacological studies, in behavioral assays, learning and memory analysis using the modern gene editing tools. I think on behalf of the organizers, Dr. Lata, the principal, Maitrey College, and my personal behalf, I would like to thank Dr. Mutkal once again for a very nice presentation. And there are a few questions.
Okay, sure.
If you permit me.
Yeah, sure.
Ma'am, the first question is, can zebrafish model serve as a good model for clinical trials also?
See, clinical trials are absolutely necessary because ultimately it's the humans who are going to, take these drugs. So we can't bypass human clinical trials. No, we can't. And that's why, what we can reduce is the other. That's why I didn't mention the strategy involving humans. We have to have a minimum number. The first clinical trial, which requires 10 to 20 human subjects, and then we test if there's any side effect or the efficacy of the drug is tested. Then it goes on to the second trial, second phase clinical trial, where around 100 to 200 people are tested, and then if it passes through the second trial, it goes to a larger population, to around 1,000 to 1,000 to 2,000 subjects. So that we can't bypass. What we can bypass is, what we can reduce is the regulatory time, the processing time by the regulatory authority. Preclinical stage we can reduce, and of course the first stage, the drug discovery and the research that goes on, involving AI and computational bioinformatic tools, where you can easily, shortlist the number of drugs by bioinformatic tools. But we can't bypass the human trials. No, we can't.
Certainly, no.
And we can't replace humans with any model system. We can't replace humans.
Absolutely. No bypassing the human trials. That is not possible.
Yeah. You see, that's why the vaccine, we're waiting and waiting for vaccine, and then, okay, this day this has been done, still there was so much controversy that, so that we can't bypass, yes.
The second question, ma'am, is please explain the difference between regeneration and healing.
Regeneration and?
Healing.
Healing. Yes. So regeneration is like, now you have like we can't regenerate our limbs. We can't regenerate what is lost in, heart, for example. Early, of course, early children, small children, they have the fingers cut, they can regenerate. Healing is just, the sealing of I'll say that you have the collagen and the keratinoid, other cells, epithelium cells, they proliferate to a certain degree, yes. And then you have a sealing of the wound, I'll say, okay. But then it will not grow further. Like, we cannot actually replace the tissue that happens in regeneration.
Ma'am,
have I answered? Yeah.
Thank you. Can zebrafish be used as a biosensor
for
river pollution? And
yes.
If yes, then what are the effects of pollution on zebrafish?
So we worked on, our first project was on Yamuna River, actually.
Okay.
And our report was really, it works, it's so sensitive, I'll tell you. In the lab, we have to be taking care of the water that we're using to grow them, especially the larvae. It's very, very sensitive to pH, to pollutants, that they won't survive. They won't survive. They will be immediately, they'll die. So we have had issues, maintaining fishes many times, and especially in rainy season when, we have a lot of acid. I don't know whether they add in the water, but then a lot of issues came up recently, and then we had heavy rains, and the water level quality really goes down, I don't know what happens, whether they're adding chlorine or what they're doing, but then fishes start dying. It's a very sensitive sensor, very sensitive, especially water pollutants, very sensitive. We can't use it for air or because naturally fishes are living in water, so the pollutants have to be dissolved in water, right? So testing of air pollutants, I won't say it's really feasible that way, but anything which is water soluble or which can be made soluble can be tested with zebrafish. It's worked as a very good biosensor, yes, for water testing, yes.
One final question: if you have a molecule in hand, how long does it take for you to do the preclinical studies on zebrafish model and tell whether the drug is actually working?
See, if you're naturally to get number of repeats and to have scientific validation, we have to have minimum number of eggs. So if we're getting a regular supply, what happens is that zebrafish can breed or lay, a female can lay eggs with a gap of 7 days. If a pair, male and female, has bred, we keep from feeding every week. So a fish is able to, the egg will grow and then lay eggs. It can lay eggs every week. So with a gap of 7 days, you can collect eggs. So that is the advantage. So if you're collecting eggs and you have a supply of eggs, the time is just one assay. And if a throughput assay, you just requires a week, just a week. But then, of course, you identify the drugs, screen them, and then if you want to go in for further molecular analysis, you want to study the assay's gene effect, then you can do a RT-PCR one. I think a few slides I missed on, maybe I forgot to add. You can do, what you can simply do is, you can collect the larvae and do a RT-PCR or a gene RNA sequencing, and you can identify the genes that are impacted. You can do that, yeah, very easily. So maybe a month or maybe that's it. If you have a regular supply of, yeah, that's it.
One final question, ma'am. A student wants to know what is the cost of establishing a zebrafish lab?
Yeah, the cost is not much. I'll say it's the most cost effective. Initially, what you just require is a room centigrade, just the AC will do in summers, and you can have a heater in winters. And another thing that one has to take care is the photo period. It follows the same photo period like we do, so they have to sleep in the night. So we have a lab where the lights go off at 9 p.m. and switch on at 7 a.m. And that's it. You just need tanks. See, for 2 years when we started work on zebrafish, we worked in tanks, in simple water tanks. You can what you do in in lab, simple tanks we bought. It's just recently, a few years back, 2-3 years back, that we have this habitat system. So you don't need that actually. If you regularly change water like you do it at home, the advantage is that we can, like many people maintain aquariums at home. It's simple as that.
Thank you so much, ma'am. Thank you so much.
We got our first pair of fishes. Yeah.
Over to you,
So one thing is, yes, one thing I want to tell you, we work on the strain ASWT that we got from IGIB. This is Assam wild type strain, which is again an indigenous strain whose genome has been sequenced, so that's why it's relevant to work with that. Is that once what your results you're getting, you can correlate because the genome is sequenced. So if you want to do work, so the first pair we got from IGIB in Mathura Road, Delhi, and then we multiplied it and maintaining that strain.
Thank you, ma'am. Thank you so much. Lata, over to you.
Thank you, ma'am, for sharing your experience and knowledge on the importance of zebrafish model system for drug discovery. And I'm sure that our attendees surely get updated with the application of this model for searching the better drug candidate. Thank you, Pasricha ma'am, for coordinating this session and sparing your valuable time from your busy schedule for this conference. Thank you, Mr. Kamal Sharma and Dr. Pooja Baveja for organizing this session. Here, with the end, the technical session 2 of the first day of this international conference. Now, we have dispersal for parallel oral presentation sessions and related details and their Google Meet links already shared with the schedule. Thank you all.
Thank you, Lata. Thank you.
Thank you, Lata. Thank you so much.
Thank you, ma'am. Thank you so much, ma'am.
Very good afternoon to all. Now the session for the oral presentation. So for each session, schedule already have been shared. So for the each session, there is a Google Meet link is there. So you all have to join by that Google Meet link. So list already have been shared. So you have to see your time and the session, and you have to join in that session only. Thank you to all.